Hello, everyone. Good afternoon.
Good afternoon, ma'am.
Good to see you. Good to see you all. Right on time. Hi, Soumya.
Hello,
Hi.
ma'am.
So, as we get started, we have people coming in. Sonal, I see you're there. You're the first speaker. Great. So, get your slides ready. Uh,
Yes.
and if everyone else can please, yeah. Yeah. Hi, Ramre. Hi, Ramre. Hi, Vinita. See you both.
Hi, hi.
Hi. And yeah, just waiting for Amit. Shala, I'll just mute you in the meantime. Yeah, just waiting for Amit to join. But yeah, Sonal, yeah, your slides, be ready. Yeah, sounds
Krishna,
good.
can you hear me?
Yes, we
Uh,
can.
Because it's
Absolutely.
raining so heavily, I'm not sure whether the sound is
Okay.
going across or not.
Really?
Okay, thanks.
Okay. Vinita, you're in Pune only, na?
Yeah, yeah.
It's raining so much at your end?
Not your end?
No.
Modi's visit is cancelled and it's
Is
raining
it? Ah,
weekend.
visit cancelled. Okay. I didn't know that. Okay. Okay.
It started
Okay.
raining quite heavily just now, about 10 minutes ago.
Okay. Not so much in our side.
I see.
Yeah. Kids have a day off from school because the visit was there.
Yes, I know, but it got cancelled this morning sometime, I think. They
Oh,
expected no crowd at SP College public meeting, I suspect. I
yeah,
suppose.
yeah,
I don't know.
yeah. All right, you settle down, sure. So we are having people come in. That's great. I have some basic introductory slides. It's going to like literally take 3 minutes, and then we will start with Sonal's talk. It promises to be a very exciting afternoon session. Five really good talks are lined up, and we hope you get all the feedback you need from the expert mentors, how to improve the talk. Sure. More people filtering in. It's 2 p.m. So let me just start with sharing my slides. Great. Yeah, day four, session two. Ooh, that's something. Rutuja, you are handling the waiting room, na, Rutuja?
Yes.
Great, great. Thank you. And Manjula, you can keep time as always. Yeah. And what will I do? I will moderate and get people into the waiting room if needed also. Okay. Hi, Arushi. Good
Hi.
afternoon. Yeah. We're just seeing each other in different waiting rooms at this point. Yeah, it's been a busy week. It's been a busy week at India Bioscience. All right, so Ramre is here, Vinita here. I don't see Amit yet. I'm sure he'll be joining. Um, but our speaker is there, Sonal. That's great. Shall we slowly get going? Yeah,
Yes, from my side.
sounds good,
Sure,
Ramre. Thank you.
sure.
Perfect, perfect. Just to respect and keep time for everyone. Great. So, welcome. Good afternoon. Hope no postprandial drop in blood sugar levels. But the science is and no better topic than medicine and physiology to make sure you understand that and still stay attentive. So, welcome to all our PhD researchers. So great to see you. And some of you have been coming consistently through the week. That is so amazing. Thank you. Thank you. So today's session is medicine physiology. We have five exciting talks lined up here, and these were shortlisted. All of you applied. We shortlisted you, and we gave sort of importance to those in the senior and advanced stages of their PhD, so you can get all the feedback you need and a pre-defense kind of experience before you go into your actual defense. So all the very best to all of you for your talks. I'm sure it's going to be great. So just some instructions on how this works. Make sure you see us, hear us, and can see slides. The chat window, feel free to keep it active. Type in your questions, suggestions, feedback. We want to hear from you there. So each PhD speaker will be given 20 minutes to talk. Okay, after 20 minutes, and we really do stick to time. After 20 minutes, there is 10 minutes of Q&A, which will be led by our expert mentors, which today are Vinita, Ramre, and Amit. If we have time after the expert mentors give you feedback, then audience questions can be taken. Else, the audience questions are in the chat window. So you can answer them directly there too. All right. So 20 plus 10 is the format, and we will need to stick to time because we have 5 talks today. And of course, feedback should be constructive, feature positive. And so we thank our expert mentors for being here and giving their time. Everyone has so much going on. You all certainly do. So thank you for giving your time at the outset. We really appreciate it. And the audience, the expert mentors feedback is there for you to listen to. The talks are there, but there's also incentive to keep the chat window active, because for audience member who keeps the chat window most active, we have a giveaway of a physical copy of our newly published e-compendium led by our colleague Ankita at India Bioscience. So, um, you know, keep the chat window active and also get a chance to get a physical copy of an India Bioscience book, and you will be the eighth recipient of this book because we've given 7 books away so far. All right. So that's that. I think we will get started now. Nothing more at my end. Rutuja, my colleague, will map the waiting room. Manjula, my colleague, will check on time. Our first speaker is Sonal Fande. Who's going to Sonal? See you, hear you?
Yes.
Yes. And you can start slide sharing, please. And
Uh,
Sonal is going to talk about biosensors, wearable biosensors, as for monitoring of biomarkers, pathogens, and processes. Sounds very exciting, Sonal. So all the very best. We can see your slides. Okay, yeah, perfect. 20 minutes, Nehal. Okay. Sorry, Sonal.
yes,
20
yes.
minutes, Sonal. Time starts now.
A very good afternoon to everyone. I'm a Sonal Khande, and today I'm very excited to present my research work on development of wearable, fully integrated biosensor for real-time biomarkers, pathogen detection in point-of-care devices. Before I begin, let me ask you a simple question. Are you really healthy? Take a moment to think again. And how do we know if you really healthy? So, many diseases are there, like cardiovascular diseases, which claim 17.9 million deaths every year. Like this cystic fibrosis is also responsible for 2.6 million deaths every year. Like this, uh, like these other diseases, many common diseases. Sorry.
Sorry. Any issue, Sonal?
Actually, I'm not seeing my screens.
We can see your screen though. What is the issue?
I'm not able to see my screen.
Really?
I'll share it again.
Okay. Try again.
Yes.
We see it. You see it? Yeah. Sonal, do you see it?
Yes. Yes. Like these other common diseases are I'm facing some problems.
Okay, Sonal. You know what? Maybe we give you a little time to figure that out,
Yeah,
uh,
yeah,
so the flow of your talk doesn't get interrupted. Our
yeah.
next speaker is Payal Gupta. Payal, I see you're here, and I I would request if you could get bumped up to being the first speaker. Is that okay, Payal? Payal, can you hear us and see us? Payal is there in the meeting room. Payal, can you hear us, see us? Okay, Payal can't hear us and see us. Next on the list is,
Hello.
yes, Payal, yeah,
Uh, yeah, ma'am, I can hear you.
yeah.
So,
Just take a few minutes to set your slides. I know we've
तेरी आवाज नहीं आ रही है
just come up on this very suddenly.
ma'am, am I audible?
नहीं
Yes, Payal, you are.
Okay, so just give me a second. I'll open the slides.
Sure. take your time. Sonal, in the meantime, you can just find out what's going on.
Yes, sure.
Thank you. Yes, Payal, we see your slides and thank you so much for taking this on. It was unexpected. Anyway, go ahead. All the best.
You're muted, Payal.
You're on mute. Yeah, it's fine now. So, good afternoon, everyone. My name is Payal Gupta, and I'm working under the guidance. I'm mute here, Sabi.
We can hear you,
We
Payal.
can
We can
hear,
hear
we can
you.
hear her
Yeah.
nicely. It's
Okay.
fine.
Yeah. So, yeah. So, good afternoon, everyone. My name is Payal Gupta, and I am working under the guidance of Dr. Ambadas Rode in Regional Center for Biotechnology, Faridabad. Today, I am going to talk about targeting G-code replexes in SARS-CoV-2 RNA genome with tetraphenylethrine derivatives for antiviral therapy. So, the coronavirus disease is a global pandemic condition that is caused by the SARS-CoV-2 virus. The SARS-CoV-2 virus contains positive single-stranded RNA viral genome. This RNA viral genome codes can form codes for 4 structural proteins and 16 non-structural proteins. The structural proteins include spike, nucleocapsid, membrane, and envelope proteins. The spike proteins helps in the attachment of the SARS-CoV-2 virus to the ACE2 receptors of the host. The nucleocapsid protein is responsible for the viral packaging, whereas the membrane and envelope proteins are responsible for the viral assembly. The symptoms for the coronavirus disease includes cough, fever, difficulty in breathing, and the pneumonia in severe cases. The SARS-CoV-2 can be transmitted from one person to another when an infected person shares the virus through coughing or sneezing. The SARS-CoV-2 replication cycle begins by the attachment of coronavirus to the ACE2 receptors of the host cell, which leads to the binding and the entry of virus into these host cells. Once the viral genome is released, the translation of viral polymerase, that is RNA-dependent RNA polymerase, takes with the help of non-structural proteins. Then the RNA replication and the transcription is mediated by this RNA-dependent RNA polymerase that leads to the replication of genomic RNA and the formation of subgenomic transcript. And the nucleocapsid RNA is translated to form nucleocapsid protein, which helps in the viral packaging. Whereas the other three proteins, spike, membrane, and envelope proteins, are translated by the help of endoplasmic reticulum, and these all combines to form a new virion. And once these virions are exocytosis, they can infect the other cells. There are different therapeutic approaches that have been used to combat with the COVID-19. So one of them is the development of vaccines. There are different methods or the different type of vaccines which have been developed. That includes the development of lentiviral vaccines, inactivated virus vaccines, adenovirus vaccines, and the DNA plasmid vaccines. But despite of different types of vaccinations available for the SARS-CoV-2, there are several limitations of the same. That includes the need for multi-dose vaccination. Their efficiency can be varied based on the doses and the type of SARS-CoV-2 variants. And one of them is the continuous evolving nature of the SARS-CoV-2. Along with the vaccines, there are several drugs, antiviral drugs that have been used to combat with this COVID-19. These drugs can inhibit the different stages of the SARS-CoV-2 replication, that can be virus binding, internalization, trafficking, and the membrane fusion. Now, because since the vaccines and the drugs are available, but still the SARS-CoV-2 variants are a major concern. Here, the wild type strain of the SARS-CoV-2 is, we can see that it is mutated, its spike is being mutated to form the different type of mutants. So one of the problem with these variants is that these vaccines and the antiviral drugs cannot act on these variants, because they might not act on that target because of the difference in the sequences. So what we can do is we can use the different structures. Here we can use, along with the protein targets, we can also use the nucleic acid targets. And there are several advantages of using the nucleic acid targets over protein targets, such as more accessibility to the target sites, more specificity, and the presence of conserved functional domain. In one of the recent studies, they have shown that the SARS-CoV-2 genome is contains different structural secondary structures, that is G quadruplexes, stem loops, and pseudoknot. Here, I will be particularly talking about the G quadruplex structures. G quadruplex structures are formed by the guanine-rich region through the Hoopstein base pairing. This leads to the formation of the G quartet, and these G quartets stack onto one another, forming G quadruplexes. These G quadruplexes can be of three types based on the orientation of the strand: antiparallel, parallel, and the hybrid G quadruplex. The G quadruplex was first, the idea of G quadruplex was first proposed in the 1962, and in the 1992, it was the first time that was shown that the G quadruplex ligands can be used to stabilize the G quadruplex structure. In 1997, the first G4 interactive compound that can inhibit the telomerase was found. In 1998, oligonucleotides with G4 structure had anti-HIV activity was discovered. In 2008, a G4 interactive helicase in SV40 was characterized. In 2009, SUD in SARS interacted with G4 had a high role in high pathogenicity. In 2013, G4 in HIV were linked with transcription and viral infectivity. Similarly, in 2014, G-force in mRNA of Ebola virus repressed translation was studied. And recently, in 2020, the SARS-CoV-2 viral genome also contains G quadruplex has been known. And there have been studies that show that these viral G quadruplexes can be targeted using the small molecules. And here's the list of the viruses that can be inhibited by that's their replication, transcription can be inhibited by these G quadruplexes ligands, such as in the case of the HIV, these transcription and replication can be inhibited by BRCA19. In the SCV, FINDAC3, TMPYP4, and pedostatin can inhibit the transcription and the replication. In the Zika virus, it's TMPYP4 and the BRCA19 which have been studied. And here's the other viruses that have been used, and the G quadruplex ligands. Now, the SARS-CoV-2 genome was mapped to identify the G quadruplex structures, plausible G quadruplex structures that can be formed. Here's the list of the G quadruplex, but the G quadruplex of this nucleocapsid gene has the higher G score and the shortest length, making it more possible, so that it has higher propensity to form the G quadruplex. So this is the structure that can be adopted by this G quadruplex. Here we have named it as RGQ1 for our further slides. This is composed of two quartets, and this is the sequence, and we have further mutated this sequence where we have completely disrupted this G quadruplexes for our biophysical assays. We further validated the RGQ1 folding into the RNA G quadruplex using the fluorescence assay. Here we have used two fluorescence ligands, thioflavin T and the NMM. These ligands are known to increase fluorescence after their binding with the G quadruplexes, whereas in the case of the single-stranded RNA, they do not show any change in the fluorescence. So here we can see that in the presence of thioflavin T, there is an increase in fluorescence intensity in presence of RGQ1, whereas in the mutant, we do not observe any significant change in the fluorescence intensity. The same experiment was performed in the presence of N-methyl mesoporphyrin, that is NMM. And here we can see that in the presence of RGQ1, there is increase in fluorescence intensity of the NMM, whereas in the mutant, we do not observe any significant change in the fluorescence intensity. We further confirm our RGQ folding using the CD spectroscopy. Since I said that these G quadruplexes can have three different topologies, parallel, hybrid, and antiparallel, their formation have different CD signature peaks. In the case of the parallel G quadruplex, there is a positive peak at 260 nanometer and the negative peak at 240 nanometer. In the case of the hybrid G quadruplex, they have positive peak at 295 and 260 nanometer. Sorry. So in the case of the hybrid G quadruplex, they have positive peak at 295 and 260 nanometer and the negative peak at 240 nanometer. In the case of the antiparallel G quadruplex, they have a positive peak at 295 and 240 nanometer and negative peak at 260 nanometer. So this is in the case of the DNA G quadruplex, but in the case of the uh, RNA, it can only adopt the parallel topology. So we confirm that this RGQ1 falls into the parallel G quadruplex using the CD spectroscopy. And here we can find that there is a positive CD signal at 260 nanometer and the negative CD signals at 240 nanometer. So this concludes that the RGQ1 falls into a parallel G quadruplex topology. Then we synthesize the small molecules to target these RGQ1 derived from SARS-CoV-2 nucleocapsid. In one of our recent studies, we have shown that these TPE derivatives can stabilizes the G quadruplex structures, suppressing the translation of TRF2 proto-oncogenes. So this is the parent compound, tetraphenyl and these are its derivatives, TPPY, MEPY, and LIPY. So here we have shown that this TPMEPY has quaternary ammonium salt that leads to the stabilization of the RNA G quadruplex. Here we have further modified this methyl ring to the allyl ring and we have formed the TP-allyl-PY. Ma'am, can I just take a second? I just need water.
Sure, sure. Please go ahead. Go on.
I'll just come in a minute.
Sure. I think it's a good time to tell everyone that the chat window can be active. Please share your feedback, suggestions. Audience members, expert mentors will directly do it, but audience members can share it. And there's really a fun giveaway happening. So you want to be in contention for that. We'll just wait a little bit till Payal is back. But in the meantime, does anyone want to drop their location? Which part of India are you joining from? I could go first. Pune. Does anyone else want to drop their location? How national is PhD Cafe? Let's find out. Bangalore. Thank you, Manjula. Hi, look at Mumbai, Hyderabad, Delhi, Nashik, Chandigarh, Oh wow, Faridabad, Delhi. Look at that. Thank you. Thanks for dropping that location pin. That's great. Bangalore, yes. Ramrev. Patna, wonderful. Thank you. So yes, I don't think we've covered all the states for sure, but
Hello, I'm sorry,
yes,
I just have to leave in
no
between.
worries. No worries,
So,
Payal. Go
yeah,
ahead.
so we further modified this methyl ring to the allyl group, and this is TP-allyl-PY, which we have studied. So this is the synthesis scheme, and just a second. Just a second. Next theory. Just a second. I'm sorry, I guess there was some problem with the slides. Yeah, so
Yeah, we can see your slides, Payal.
Yes. Okay, ma'am. Yeah. So this is the synthesis scheme for the TP derivatives. So we have further screened these derivatives binding against the RGQ1 using IMSA. Here we can see that in the presence of TP-MEPY and TP-LIPY, there is a presence of the retarded band, whereas in the presence of TP and TP-PY, we do not see the presence of the any retarded band. That means these TP-MEPY and TP-LIPY interacts with the RGQ1. Then to further confirm the specificity of these ligands, we perform the same IMSA experiments in the presence of mutant RGQ1M. And here we can see that in the presence of RGQ1, there is presence of the retarded band. But in the same gel, we can see that in the presence of mutant, we do not observe the presence of any retarded band. Then we further confirm this binding using the fluorescence assay. Here we utilize the intrinsic fluorescence property of the TP derivatives, that is TP-MEPY and TP-LIPY. We can see that in the presence of RGQ1, there is an increase in the fluorescence intensity of the TP-MEPY, whereas in the mutant, we do not observe any significant change in the fluorescence intensity. Then we perform the same experiment in the presence of TP-LIPY, and we can see that in presence of RGQ1, there is decrease in the fluorescence intensity of TP-LIPY, whereas in the mutant, we do not observe any significant change in the fluorescence intensity of TP-LIPY. This contrasting behavior of TP-LIPY could be due to the different binding mode. So, in conclusion, we conclude that TP-MEPY and TP-LIPY specifically interacts with the RGQ1. Then, to determine the thermodynamic parameters of binding of TP derivatives with RGQ1, we did the isothermal titration calorimetry, that is ITC. And we can see that these TP derivatives bind to RGQ1 in 1:1 binding mode. The KD for TP-MEPY was 0.274 micromolar, and for TP-LIPY it was 0.232 micromolar. The delta H was negative, that means the reaction was exothermic, and the T delta S was positive, suggesting the favorable changes in entropy. So next we check that does these compounds TP-MEPY and TP-LIPY stabilizes the RGQ1 structure. So here we perform the CD analysis, and we can see that in presence of TP-MEPY, there is increase in CD signals at 265 nanometers, whereas the same could also be observed for the TP-LIPY, where in the presence of 25 micromolar TP-LIPY, we see an increase in the CD signals. Then to quantify that, what is the change in the TM of these RGQ1 in presence of these two ligands, we perform CD melting, and we can see that in the presence of TP-MEPY, there is increase in TM by 8.54 degrees Celsius, and in the presence of TP-LIPY, there is increase in TM by 12.56 degrees Celsius. Then we perform luciferase assay to check that does this stabilization suppress the translation of the luciferase gene. So we clone the RGQ1 and its mutant in the 5' UTR of the ranula luciferase gene. Then we co-transfected this clone plasmid along with our normalization control plasmid, where we have used the ECGFP. And here we can then first we seeded the cells, we co-transfected the reporter and the control plasmid. After co-transfection of these plasmids, we did the compound treatment, and after lysis, the readings were taken. Here we have considered the luciferase activity to be 100% in the presence of DMSO. So here we can see in the presence of TP-MEPY, there is suppression of the luciferase activity by 2.2 fold in presence of MEPY, whereas in the TP-LIPY, we see suppression of the luciferase activity by 3.6 fold. And in the presence of the mutant, we do not observe any significant change in the luciferase activity. This suggests that TP-MEPY and TP-LIPY stabilizes this RGQ1 structure, which leads to the suppression of the luciferase activity. Then, to determine the antiviral activity of these TP derivatives, we performed the immunofluorescence assay in Vero E6 cells. For this, we did the cell seeding, and after 24 hours, we did the compound treatment. After 1 hour of compound treatment, we did the virus infection, and after 30 hours of virus infection, we did the cell imaging. Here we can see that in the presence of TPMEPY and the TPLIPY, the cells are viable, but there is a decrease in the nucleocapsid level by 63.26% in the case of TPMEPY and 77.9% in the case of TPLIPY. Then, to further determine the mechanism of action of these TP derivatives, we adopted two different strategies, pre-infection treatment and the post-infection treatment in the A549 cells. So here we did the post-infection treatment. For this, we did the cell seeding of A549 cells. Then the virus, SARS-CoV-2 virus was infected at 0.1 MOI for 1 hour. And after 1 hour, the cell culture media was replaced and the compound treatment was performed for 48 hours. After 48 hours, the cells were harvested and we performed the RT-PCR and the Western blot assay. In the RT-PCR, we can see the SARS-CoV-2 RNA level is decreased in the case of TPMEPY and the TPLIPY. Here we have also used a G-quadruplex ligand, very well-known ligand, that is predostatin, that also shows the same result. Then we check that what is the SARS-CoV-2 viral RNA level in this viral supernatant. And we can see that in the case of the in the viral supernatant as well, the TPMEPY and TPLIPY is decreasing the viral RNA level. That means the viral progeny is being inhibited. We further confirm this data using the Western blot, and we can see that in the presence of TPMEPY and the TPLIPY, there is decrease, a suppression of the nucleocapsid and the spike levels. That means our RT-PCR and the Western blot data suggests that these TP derivatives act on the post-entry stages of the SARS-CoV-2 replicative cycle. That means they are stabilizing the nucleocapsid gene, G quadruplexes, and inhibiting the viral RNA level. Then, to determine that does these compounds also affect the viral entry or the pre-entry stages of the virus? So here we did the pre-infection treatment in the A549 cells. We did the cell seeding for 24 hours. After cell seeding, we did the compound treatment for 1 hour. After 1 hour of compound treatment, we did the virus. We infected the cells with SARS-CoV-2 at 0.1 MOI for 1 hour. After 1 hour, this cell culture media was replaced, and the cells were further incubated for 48 hours, and then the cells were harvested. And we can see that in the presence of TP derivatives, we do not see any significant change in the viral RNA level. Whereas in the Western, similarly, we further verified it with the Western blot data that these derivatives do not affect the do not show any change in the nucleocapsid and spike level in the pre-infection treatment. These suggest that these TP derivatives, that is TPMEPY and TPLIPY, they do not affect the early stages of the viral replication cycle. That means it does not inhibit the viral entry, viral attachment, and the endocytosis. So in conclusion, the SARS-CoV-2 can interact with the ACE2 receptors, and once the binding is done, there is the release of the viral genome. That means our compound does not act on these entry stages. And once the viral genome is released into the host cells, there is the formation of the RNA G-quadruplex structures in the open reading frame of the nucleocapsid gene, which is stabilized by these TP derivatives, further inhibiting the RNA replication and the subgenomic transcription, which further subsequently reduce the formation of new SARS-CoV-2 variant. This data has been, we have submitted this manuscript and is currently under revision. So at last, I'd like to acknowledge my supervisor, Dr. Ambadas Rode, for his constant support and guidance. Our collaborators, Dr. Milan Sujit and Dr. Sudhanshu Brati, for the antiviral assays. Dr. S.V. Bhosle for synthesizing the compounds. RCB and CIF, my lab members, CSR for fellowship, and India Bioscience for providing me an opportunity to present my work here. Thank you. And now I'm open to the questions.
Excellent talk, Payal. You made it. We made it. Technical glitches and everything included, but thanks for coming one step higher in the order of the speakers. I know that caught you a little suddenly. So Ramre, Vinita, Amit, Amit, great to have you. I think you joined a little late, but I know that you're there. So do share your thoughts with Payal. We have 10 minutes for feedback. Sure.
Maybe I could go ahead, Karishma. Am I audible? Payal, am I audible to you?
Yes, sir.
This is Ram Rai here. Excellent talk. Very comprehensive work. I just had a couple of questions. Um, Payal, do if this is these are agents that stabilize the G quadruplexes, how do they perform vis-a-vis other several other G quadruplex inhibitors that you showed in your second slide? In other words, have you benchmarked your inhibitor with other G quadruplex inhibitors that are there in the market and so on?
Since right now only one of the G quadruplex ligand is in the clinical trials, whereas
Mm-hmm.
others are just commercially available.
Mm-hmm.
So if we talk about the G quadruplex ligands, they can be varied based on their mechanism of action as well as the selectivity based on their KDs. So that could be one of the way that we can separate our ligands or differentiate our ligands with other G quadruplex ligands.
Okay.
Here in we have used the pedostatin as a positive control, which is a very well known G quadruplex ligand, and here we have found the similar results. But I hope that maybe these compounds will have better penetration and other
Sure.
cytotoxicity as well.
Sure. That was what I was coming towards. I'll just be brief. There are just a couple of suggestions. If this is like if you are at a revision stage of your manuscript, or in terms of writing thesis, you might want to think about commenting on the use of A549 cells. These are, after all, lung cancer cells, right? So the
Yeah.
adenocarcinoma cells.
So
Entry kinetics, the cytotoxic kinetics will all be very different. You could perhaps just keep a caveat for this in whatever you write up in the future. I know
yeah.
that doesn't make sense to tell you, oh look, let's do something else,
No,
or lung
nothing
organoids
like
and
that. But the thing is that we have used the A549 cells because of the two reasons. First of all, it is in the lung cancer cell line, and the SARS-CoV-2 mainly affect the lungs. And so most of the studies which have been previously published, they have used the A549 cell lines or the VROE6 cell lines. So we have
yes.
used both of them.
Sure. Like I said, lung is not lung cancer, right? So that's
Yeah,
and
understood.
I'm being a cancer biologist, have to mention that. But
Yeah,
nevertheless, like I said, that's why I said just keep this as a comment in your
yeah.
ideas, or in terms of your other
Hmm.
assumptions that you make in your book. I'm done. Thank you so much. Good luck for the future, Payal.
Thank you, sir.
Great. Thanks, Ramre. Amit, Vinita.
Uh, shall I go ahead?
Yes.
Vinita. Yeah. This, I think Ramre has already given more technical input than I can possibly give, but I'm going to comment on the general presentation as well, because that's also something that you will require. So, Pile 2, 3, 4 different things. One is, I think you spent too much time in the introduction. You took about 7, 8, 9 minutes before you arrived at the data. For a 20-minute presentation, 20-minute talk, spending more close to half the time into introduction and background is not a great idea, because your emphasis should be on the data that you are presenting. So keep that in mind.
Yes.
I also realized that when you are talking about G quadruplex history, you showed a long list of which viral infections it is useful, effective, and so on and so forth. How much of that is needed is something that you should think of, because what we are here and what we are going to judge you for is your data primarily. So
Yes.
think about it. It's not completely a negative point, but I wanted to mention this since you will be presenting data more often. So looking at the time limit, you have to plan what you will show and where you will put more emphasis. The second point, for old people like me, having very small font is a detractor. at many places I couldn't read what you were saying, especially this amino acid or this nucleotide or that nucleotide. Keep that in mind. I know I have been very insistent about this when I was a PhD mentor, no longer, but that's something also that is useful. So how much data will you show and how much data can you convey to the importance of that data can you convey to the audience so that audience is with you? If you have very small font and if you are going very rapidly, people like me will get lost. All right. So these are the general comments. One comment that I want to ask you about is that between your TPE, MEPY, and LLPY, there was one slide where they behaved differently.
Yeah, in the fluorescence assay.
And on the rest of the assays, they are behaving quite similar to each other. What is the reason you think as for the difference?
So in the other studies, we have just characterized here it is the fluorescence behavior that is being different, whereas that could be because of the difference in the binding mode. But if we talk about the other assays, these are the biophysical assays that shows that how does this affect the stabilization and its functions. So maybe the binding mode is different, that is leading to the change in the behavior of these compounds.
So,
but their
okay,
functional
so
activity is same.
Sure. So does that mean that your assay, fluorescence assay that you are using needs a modification as a general rule? Because they gave you different, the assay gave you different results and functionality is similar.
Yeah.
How would you modify that?
Maybe that could be, we can say here we use the mutant as well to confirm the specificity. So
you have controls. I'm not commenting about the controls. I'm commenting about whether the assay is, meaning fluorescence assay or the compounds that you're using in the fluorescence assay need a second look. Because there are differences that you are observing, and then there are differences which you are not observing in the two compounds.
Yeah, maybe we can look up to it. That was, yeah.
Okay. So you showed a preventive assay and you showed a therapeutic assay, right? Prevention with the compounds, and you added first gave infection followed by your compounds, right?
Yeah.
So
yes,
why did you use different assays for one versus the other?
So the post-infection treatment here, it is that we did the virus infection first, and then we did the.
No, I got the methodology. I'm asking about
so
the results
the
that
post-infection treatment was done to check that it affects the post-entry stages of the viral replication cycle. Whereas in the case of the pre-infection, it shows that does it affect the viral entry or the early stages of the replicative cycle, that is before entering of virus to the host cells. That is viral attachment, viral entry as well.
Okay. So my last comment, which is very positive, is your slide, concluding slide was very good. Thank you.
Thank you, ma'am.
Sounds great. Amit, nice to have you. Yeah.
Great. Thanks. Thanks, Reshma, for having me here. So great talk, Payal.
Thank you.
That was kind of like well presented. I was just intrigued with the specificity of your TP derivative, I think it's the MEPY to the G-cortical cells. I was just wondering if you would already be knowing if the other regions or other RNA structures and all it could be binding to as well, or if you can comment more on what would be the exact or some kind of like binding mode. How does it really bind? Does it bind to the phosphate backbone, or does it bind to the
So
base
basically,
itself?
if we talk about the G quadruplexes, as I showed the structure of these G quadruplexes, there are the presence of the quartets.
So it is the quartet basis that it
So
directly binds
this
to.
is the quartets. So here the compounds can bind to the between the quartets and along with this position as well. Most of the G quadruplex ligands binds here in the between the quartets.
Okay.
Yeah.
And what does it do? It induces some kind of a conformational changes in there, or
This
it is just
stabilizes the
stabilizing.
They are stabilizing the G quadruplexes.
The existing state.
Hmm.
And what really brings in the specificity?
Sorry, sir.
What really brings the specificity to?
So here, if we talk about the specificity of the G quadruplex ligands, here the specificity means it's differentiating behavior towards the single-stranded RNA and the G quadruplexes. So this is that it is selectively binding to the G quadruplexes and stabilizing it. It does not affect the single-stranded RNA or the viral other structures.
So no other RNA structure or protein that it would be binding to?
No, sir. Unless until it is containing any G quadruplex.
Okay. And how do you really envision? I mean, do you envision that this is going to be in combination to the chemotherapy kind of like, or other different kind of like treatment which is already in there, that it would have its implication? Or do you think this would be something in its own, in its absolute, it would be something that would have its significant impact?
So right now I can't comment that how long this compound will go because we have to do the PKPD study as well in the mice. So
what I meant to say was that would it kind of augment the effect of chemotherapeutic agent if it is already present? Or
so if we talk about the cancer, so
yes, in
there are
particular.
various G quadruplex ligands that can stabilize this and can affect the decrease the proto-oncogene level by the suppression. So then that could be by like for the telomerase inhibition and other mechanism are there. So yes, these ligands can be used for the cancers as well.
Yeah, I mean, that's what kind of like slightly, but in general, I mean, my last question is like, how do you really envision to translate this, if you really have to, into the clinical trials, and what would be the strong potential therapeutic implications, applications of
So
this?
so we can make the more like derivatives to check that how does the first of all we'll check its PKPD studies in the mice and then we can make the more of the derivatives that which of these ligands is better for the use. So for the clinical I can't comment right now.
I mean, eventually, perhaps, I mean, this sounds promising. But anyway, a great work. Congratulations to you and to your PI. Thank you.
Thank you, Pai. Thank you, all three of you, Amit, Vinita, and Ramre. Great. We'll move to our next speaker, which is Tanvi Sharma, who's going to talk about short chain fatty acids for metabolic syndrome. And Tanvi, you can start sharing your slides. You have 20 minutes, Tanvi. We will stick to time. And after Tanvi, we are coming back to Sonal. Sonal, please make sure slides and tech is ready. Okay, Tanvi. All the very best. 20 minutes. Great.
Am I audible?
Yes. And visible. And slides are visible. You can go into slide share mode. Great. All the best.
Thank you, ma'am. Good afternoon, everyone. I'm Tanvi Sharma, here to present my PhD work entitled Short Chain Fatty Acids as a potential therapeutic agent against high-fat diet-induced metabolic syndrome under the supervision of Dr. Naveen Kaushal. So firstly, what is metabolic syndrome? Metabolic syndrome is a complex disorder which is characterized by physiological, biochemical, and metabolic complex diseases which is caused by a cluster of interrelated metabolic abnormalities like stroke, diabetes, hypertension, and other health conditions. So the diagnostic criteria for the metabolic syndrome slightly varies and is a combination of factors like abdominal adiposity, hypertension, hyperglycemia, elevated levels of triglycerides, reduced levels of high density lipoprotein, and others. So the gut, there are these method physiology of the metabolic syndrome is also depends on the factors like genetics, lifestyle interventions, hormonal imbalance, gut dysbiosis. So the currently available therapeutic approach
Hello, Tanvi.
Ma'am, am I audible?
Yeah, now you are.
Sorry, there is a light problem. The Wi-Fi got lost. So I will continue. So the currently available therapeutic approaches includes lifestyle interventions like healthy diet, exercise, and so on. Uh, from a pharmacological viewpoint, medications targeting diabetes, obesity, dyslipidemia, hypertension includes metformin and statins, angiotensin converting enzyme inhibitors, angiotensin 2 receptor blockers. But these medications give only temporary symptomatic relief and have side effects like in metformin they leads to gastrointestinal distress. Statins cause muscle pain and liver enzyme abnormalities. Angiotensin 2 receptor blockers leads to low blood pressure. The gut microbiota, the role of gut microbiota have been implicated in the development of metabolic syndrome through its involvement in energy metabolism and nutrient absorption. Therefore, it might have a clinical outcome in metabolic syndrome. So currently the gut microbiota based therapeutic approaches which are currently being used are prebiotics, probiotics, and fecal microbial transplantation. But these also pose some challenges and risks. So like in prebiotics and probiotics, although they are considered safe, but may have non-specific effects with allergies, infections, and antibiotic resistant gene transfer. In case of fecal microbial transplantation, there is a risk of potential harmful bacteria and the adverse interaction with the host existing microbial population. So the literature suggests that the postbiotics such as short chain fatty acids are emerging as a potential therapeutic approach. So short chain fatty acids are monocarboxylic acids which are produced by the gut bacteria through the fermentation of dietary fibers and polysaccharides in the colon. It plays a crucial role in immune defense, digestion, intestinal, maintaining intestinal homeostasis, maintaining a healthy gut. So there are various short chain fatty acids produced, but they primarily are acetate, butyrate, and propionate, mainly in the ratio of 3:1:1, in which the acetate is the most abundant short chain fatty acid that is produced in the body, which is produced by a variety of bacteria like Bacteroidetes and Firmicutes bacteria. Propionate is produced by Bacteroidetes and butyrate is produced by Firmicutes bacteria. It is important to note that the diet that is what we eat strongly influence and it is a crucial role in synthesizing the short chain fatty acids in the gut. So considering that the experimental design was established with male Vistar rats, dividing into 4 groups after acclimatization for 1 week. The control group was given normal pellet feed for the entire duration of the experiment. Second is the high-fat diet group, to which high-fat diet consisting of 2% cholesterol, 1% of cholic acid, 47% of normal pellet feed, 40% of sucrose, and 10% of peanut oil was given for the first 8 weeks. After successful establishment of this model, it was followed for another 3 weeks. Third is the short chain fatty acid group, to which for the first 8 weeks, normal pellet feed was given, followed by normal pellet feed and a mixture of short chain fatty acids, that is sodium acetate, sodium butyrate, and sodium propionate for the rest 3 weeks. And the treatment group consisting of firstly given the high fat diet with tap water for the first 8 weeks, followed by high fat diet with the mixture of short chain fatty acids in the drinking water for the rest 3 weeks. These are the objectives which I will covering in this presentation. So coming to the results, firstly, the body weight is one of the initial indicators that needs to be evaluated, marked, and as it also depends on the food intake and the calorie consumption. So the high-fat diet rats that appears in the red color showed an increase in the body weight gain as compared to the control and SCFA alone groups. But the supplementation of short chain fatty acids, however, decreases the body weight. This suggests the effectiveness of the short chain fatty acids that improve the satiety and also the regulate the energy metabolism. Followed by the fasting blood glucose levels, which were measured by a commercially available glucometer after 8 weeks for the establishment of the metabolic syndrome model and at the final stages of the model. So the obese rat that was the high-fat diet showed an increase in this level in case of a high-fat diet group as compared to the control. But the fasting blood glucose levels were comparable to the control with the supplementation of short-chain fatty acids. Also, the oral glucose tolerance test was conducted to evaluate the glucose metabolism before the initiation of the model and after the completion of the model. So the rats post glucose administration and glucose solution, they increases with the 30-minute and gradually return to the baseline level respectively. Therefore, no significant changes in all the groups were marked with the area under the curve analysis. But after the treatment, the completion of the model, the control and SCFA alone group gradually increases with 30 minutes and return to the baseline by 120 minutes. But the high-fat diet group rats did not resolve even after 120 minutes. But the supplementation of short-chain fatty acids, however, the blood glucose levels returned to almost the low levels by 120 minutes. Therefore, the significant difference was seen with the high-fat diet and the control group, and the supplementation of short-chain fatty acids, however, reduces the and improves the glucose metabolism. Also, the adiposity was measured with the different fat depositions, which is contributed to the metabolic syndrome. So, like peritoneal fat, which is around the abdominal region, epididymal fat, which surrounds the testes, mesenteric fat, which is around the intestine, and inguinal around the groin area. These all fat resulted in the increase in the fat deposition with the high-fat diet rats as compared to the control rats. But with the supplementation with short-chain fatty acids, it was seen that the elevation in these fat depositions were observed with the supplementation of short-chain fatty acids. Therefore, by targeting the adiposities, these fat depositions, the short-chain fatty acids emerges as a promising candidate for the therapeutic potential for the regional adiposity, which offers a multifaceted approach to alleviate the metabolic abnormalities, complications which are associated with metabolic syndrome. Further, the body mass index, that is the BMI, was measured with the body weight and the length, that is the height of the rats, in which it was observed that with the 8 to 12 week, an increase in the BMI as compared to the control, but the supplementation of short-chain fatty acids, however, restored their BMI levels and were comparable to the SCFA and controlled rats. Also, the Lee index, which tells us the overall body mass and the adiposity, in which if the value is less than 3, it is considered to be obese, to have obesity. Therefore, it was observed in the case of high-fat diet group with the 8 to 12 week as compared to the control and SCFA group. But the supplementation with short-chain fatty acids, however, resolves this Lee index value. Further, to study the lipid-lowering effects of short-chain fatty acids with the high-fat diet-induced metabolic syndrome, the lipid profile consisting of cholesterol, triglycerides, high-density lipoprotein, low-density lipoprotein, and the ratios were analyzed after the 8 weeks for the establishment of the model and at the final week completion of the model, in which the high-fat diet group showed increase in the levels of cholesterol, triglycerides, and high-density lipoproteins as compared to control. But the high-density lipoprotein, which is considered as a good cholesterol, reduces these levels in the high-fat diet rats as compared to the control. But the supplementation of short-chain fatty acids, however, restored these lipid profile values, and therefore this lipid profile clearly suggests a therapeutic approach for short-chain fatty acids that can regulate the lipid metabolism. These are the morphology of the livers on the different groups, in which fatty liver is one of the surrogate markers of hypercholesterolemia. So the hepatocyte, the tissue appears, which leads to fat accumulation, leads to hepatic cytosis, and appears light yellow, palish color in color. So it was observed in the case of high-fat diet rat's liver, as compared to control and SCFA alone groups, which were deep red in color. But the advancement in the liver morphology was observed with the supplementation of short chain fatty acids. Following this, the wet weight of these livers were also measured, which is an indicator in which the high fat diet showed an increase in the weight of these wet weight livers and the body weight ratio as compared to the control. But the supplementation of short chain fatty acids, however, significantly reduces this. Following the serum aspartate aminotransferase and alanine aminotransferase activities were measured as a hepatic toxicity markers, as these enzymes are modulated more prominently after a hepatic injury. So the rats which were fed with high fat diet showed an increase in this activity and as compared to the control group. Therefore, with the supplementation of short chain fatty acids have restored the potential activities of active these levels in the ALT and AST activity, suggesting a potential hepatoprotective effects. Further, the blood pressure of the rats were also measured using tail cuff method, which helps to identify if there is hypertension, which is a key factor for to measure if any developing of cardiovascular diseases due to metabolic syndrome. So the rats which were fed high-fat diet showed an increase in the levels of the blood pressure as compared to the control and short chain fatty acid alone groups. But the administration of short chain fatty acids, however, reduces the blood pressure levels and return to the almost base of the normal levels. Therefore, this might have a beneficial effect of short chain fatty acids on the gut health and metabolism. These are the panel of photomicrographs of liver parenchyma at 20x magnification of different groups. The histology of the control group showed the hepatocyte architecture to be normal with no fat accumulation or lipid depositions and no inflammatory cell infiltrations. Whereas in the case of high fat diet, it was observed that there was hepatocyte degeneration with these gaps showed the lipid fat accumulation in the hepatocytes and inflammatory cell infiltrations. SCFA shows the similar histopathology as that of the control with no fat accumulations. Advancement in the histopathological analysis were observed with the administration of short chain fatty acids as preservation of the hepatocyte architecture and with reduced fat and lipid accumulation in the liver. Also reduces the inflammatory cell infiltrations. The redox modulation of the liver tissue was observed in which the oxidative stress is a paramount importance in physiological and pathological context due to its role in cellular hemostasis and disease progression. So in the context of high-fat diet, induced metabolic syndrome, the different antioxidant and enzymatic and non-enzymatic antioxidant markers of the oxidative stress was analyzed to assess the role of the oxidative stress in regulating the metabolic imbalance. So in case of high-fat diet, a notable decline was observed in GST, GPX, and GR as compared to the control rats, indicating a compromised antioxidant defense system in high-fat diet-induced metabolic syndrome. However, the catalase, the redox ratio, and NO levels activities in the high-fat diet animals were significantly increased, indicating the accumulation of free radicals and heightened the oxidative stress. Therefore, the supplementation of short chain fatty acids, however, effectively restored these activities and were comparable to the control groups, showing a potential antioxidative role. Further, the LPO levels leading to the MD accumulation, MDA increase, also in the high-fat diet group. Also, because of the ROS accumulation because of the disease in the high-fat diet group, an increase was observed as compared to the control group. But the supplementation of short chain fatty acids, however, reduce the LPO levels and the fluorescent intensity in TCF points with the supplementation of short chain fatty acids in the treatment group. marking the antioxidative effectiveness of the short chain fatty acids in elevating the oxidative stress linked with metabolic syndrome. It was also measured in the tissue of heart tissue, in which a similar pattern was observed, but a decline in the case of catalase was observed as compared to the in the high-fat diet group with the comparable to the control group, which can be unlike liver, the heart does not have the primarily role to break down fats. So um, this diet can lead to build up of the fat accumulations and causing damage to the liver cells, heart cells. Therefore, less able to defend itself and leading to stress. But the supplementation short chain fatty acids have restored these values. Coming to the summary and conclusion. Metabolic syndrome is a complex cluster of diseases which can be due to genetics, the lifestyle interventions, the factors, and the consumption of the high calorie diet, which can lead to weight gain, insulin resistance, dyslipidemia, hypertension, and inflammation. So the currently available therapeutic approaches like short chain fatty acids, which are produced by the gut bacteria by fermentation of the dietary fibers in the colon, mainly acetate, butyrate, and propionate, inhibits and helps to in the weight management, improves the insulin resistance and enhance the insulin sensitivity with improving genesis, adiposity, lipid profile, reducing the hypertension and inflammation, which further leads to reducing inhibition of the redox stress, oxidative stress, by decreasing the free radicals and ROS in the body, further inhibiting, protecting the liver. Therefore, the short chain fatty acids is a rebalance to the gut microbiome, and this restores the metabolic equilibrium. Therefore, this research is a promise avenue for metabolic syndrome management. These are the references. For the future, currently working on the gut-brain axis, in which to study the mechanism of how the gut microbiota and brain, there is the role in with this, and also the lipid metabolism. Further, we are planning to work on the solute transporters, how particular these transporters are involved in in amelioration of the high-fat diet-induced metabolic syndrome with the help of short-chain fatty acids. I would like to acknowledge Punjab University and the Department of Biophysics. Thank you.
Okay. Thank you, Tanvi, for making us aware about our diet. It was really interesting talk. I now hand it over to expert mentors for suggestions and feedback.
Hi, Rutuja. Can I go first? Ram Rai here. Just wanted to ask, how are we doing in terms of time? How long was this talk for? I have a feeling this.
Yeah, Manjula was keeping the time.
It was 20 minutes. It just got over on time.
Okay.
Oh, okay. All right. Fine. I just had a feeling that it went to 25, but anyway. All right. Feedback from my side is that, I had a question.
Yes, sir.
Yeah. Hi, Tanvi. Yeah, very nice, very comprehensive sort of physiological study. I had, one sort of largish query was that, if the SCFAs are coming essentially from the gut bacteria, which are able to convert this, synthesize these SCFAs from the high fiber diet, would it then be better to actually have the whole probiotic prebiotic thing that you were sort of critiquing in the beginning? In that sense, we are already with putting in probiotics or prebiotics, we will be able to generate the same SCFAs, right?
No, sir. Mainly these postbiotics are the metabolites for the beneficial bacteria. In the case of leading to metabolic syndrome, which primarily starts from obesity, and that in that cases the body does not the concentration which is produced in the body, the SCFA concentration, it reduces by marginally. So therefore, we are supplementing it in drinking water.
I see. Okay. My other sort of suggestion, more of a suggestion here is that, your first graph did not have error bars, which was your first graph where you were talking about, you don't need to go back, it's fine, where you were talking about I think the increase in body weight. In general, also in these kind of studies where you're working with mice, best to replace, what are they called? Sorry, bar graphs with points where each point represents a single animal. Increasingly journals are going towards a point where we see the spread of the data as well as the distribution of these data points, rather than just looking at mean versus standard deviations or standard errors. There were quite a few graphs I felt where I could not see the significance. Maybe you have it at the end. I started realizing that you have these figure captions where you're mentioning the significance, where it was not mentioned in the graph itself. So these are just things that could be remedied. Yeah, I'm done. I think this was also just one point. I didn't think you sort of framed the question of the study very well. You went directly from critique into experiments. If you can just frame like what's the question you're asking, and then break it down into objectives, which you also skated over very fast. You didn't mention the objectives. There was a list that you did not talk about. So do focus more on that than the sort of lengthier introduction. I'm done. Thank you. Good luck, Tanvi.
Thank you.
Shall we follow the same pattern? Should I come in now? Okay, this is Vinita Bal. I think since I commented on the structure of the presentation in the earlier case also, let me say that not only you finished your talk in time, but I thought the distribution of time was much better in your case. So I'm very glad for that you managed to do that. So a couple of questions at least. And also, as compared to earlier, most of your figures were readable. An occasional one where I could not figure out, especially when there was an inset in the blood sugar level changes that you showed. So that was something. So do you show less data but readable data, or do you show more data which is non-readable? That's a question that all of you should ask. It's not only directed at you, Tanvi, but that is what I would like to stress in general, that people should be with you when you are presenting any data. So first is, you said you gave these SCFAs in drinking water, right? Yes, so if you are, obviously you are doing this work because there is a long-term therapeutic human treatment that is in your mind, I presume. So how do you think SCFAs can be given to human beings by drinking water?
Yes, ma'am.
So
currently with the experiments, we have seen that only in the drinking water we can reduce and manage the the lipid metabolism, the weight. Weight is the initial indicator. If it is managed, many factors follow that process. So it can be given.
Okay, so this is simple oral route that you are thinking, and here 8 weeks of development of obesity and then 4 weeks of coming down with treatment. That's what your time scale was, correct? So, okay, so one question is, you mentioned acetate to propionate to butyrate ratio of 3:1:1, which is potentially ideal for the effects that you see. Am I right?
Yes, ma'am. In the body, mainly these three are present in with 3:1:1 ratio.
Okay, and then your treatment therapy, whatever molarities that you showed, did not adhere to 3:1:1.
Ma'am, that is produced in the body. Ma'am, it is close to 3:1:1. The paper that I followed, they are giving in the drinking water. Ma'am, there are many routes which short-chain fatty acid administration are done. So in the drinking water, we are trying to, these are close to 3:1:1 ratio. So we are trying, if this ratio can, as a therapeutic approach for this disease.
How expensive are these SCFAs to give?
Expensive, in just 100 for 500 grams of talking about propionate, it is only about 800
Okay,
rupees.
not expensive. Okay, let me ask you one question which is completely gone unaddressed. All your experiments were on male rats. What will you expect difference if you had used female rats?
Yes, ma'am. Ma'am, because of the hormonal imbalance, the estrogen, progesterone, they involves.
But men have testosterone.
Yes,
A
ma'am.
lot of
But in the
testosterone.
case of females, ma'am, our lab have already worked for different model, but with the female rats, so they showed a very involvement of the estrogen and progesterone. So, metabolic syndrome is more inclined to females than the males.
Exactly.
I would like to address that. So, because of technically reduced can the financial support to detailed study for the hormonal imbalances which are more in females, so we chose males, so there is no much involvement of the hormones.
I understand that rats do not have the same cycle, hormonal cycle as humans have, but you might be aware that there is a push for the scientific community to use animal models, and where animals of both sexes, males and females, are used because of the past where only one sex was used and the results were not replicable when it went to translational stage. So this is something that is worth keeping in mind, and I'm suggesting that only a critical experiment that you could do where you could have all females but with male controls, because your male control should work and females should also show. That might be worthwhile. It might add a lot of value, because this is something that is getting more and more importance from the people who are reviewing your manuscripts and are publishing work in general. Thank you very much.
Okay, ma'am. Thank you, ma'am.
Okay, we are little over time. So can we have a quick question from Amit?
Right. So Tanvi, well, most of the questions have already been kind of like, maybe I can ask like more of like a broader, this was an interesting and intriguing kind of a presentation. So if any other aspects of the gut and brain axis that warrant further investigations and that would lead to how it would lead to the next steps in this field?
Sir, I don't have that knowledge right now, but we are planning to explore this study in that area. But currently, I have not touched this part.
Okay. Or do you know of any neural disorders that are likely to be influenced by this gut microbiota derived SCFAs?
Sir,
Currently, is there?
to be frank, but with the neural, any part of the brain, what kind of disease, I'm not aware right now.
So there is no specificity to the CNS or to any specific region of the brain, right?
Pardon?
There is no specificity to any specific region of the brain. It could be
region, sir, I'm not aware of that right now. That what part of the brain we need to target and what all that can be done. I'm not aware of that right now.
And if you have to develop into a personalized medicine approach in treating CNS disorders, do you think that this gut-brain axis could contribute to it?
That might. So gut-brain axis, there are a lot of studies which are currently working on the gut-brain axis. However, how this approach leads for the betterment of that, we'll have to see.
Right. Okay. Yeah. Thanks. Nothing else.
Okay, thank you, Tanvi. Our next speaker is Sonal. Sonal, can you please confirm that you are ready?
Yes, I'm ready.
Okay. You can start sharing your slides.
Yes.
Okay.
Yes. A very good afternoon to everyone. I'm Sonal Khande, and I'm presenting my research work on development of wearable fully integrated biosensor for real-time biomarkers, pathogens, detection, point-of-care devices. Before, before I begin, I would like to ask a simple question. Are you really healthy? Take a moment to think again. And if we are healthy, how we will know if really we are healthy or not? So most of us, we do the routine checkup for the detection of various diseases. But many diseases like cardiovascular diseases, which claim 70.9 million death annually, along with the cystic fibrosis is the another cause for the death. Every year, 2.6 million people die because of cystic fibrosis. Again, other common diseases like kidney diseases, diabetes, and various infectious diseases contribute to global mortality rate. So why it is happening? The main problem is we don't have early detection techniques, and if we don't detect these diseases in the early condition, cause the severe condition, and there we don't really know which disease is growing. So here the role of biomarkers play an important role. So biomarkers is the indicator for any progression of diseases. So biomarkers are categorized into four types: DNA-based, protein-based, lipid-based, and sugar-based, which have multiple advantages in the early detection and prevention of diseases. But how will measure the biomarkers level? So there we need the device which is biosensor. So biosensor is the device which convert biological signals into electrical signals. So imagine now if we could detect our health data in at the convenience of our home also and at any time and anywhere. So that will be the better way for the tracking of our health data. So this is what we are aiming for the wearable biosensors. So biosensors are categorized into invasive and non-invasive. Non-invasive biosensors are further categorized into ocular, epidermal, and oral cavity. In ocular, the fluid is tear, epidermal skin interstitial fluid and sweat, and oral cavity saliva. So these are the four different elements of biosensor. First is analyte. So here the analyte can be a bacteria, proteins, amino acids, anything which analyte we have to detect. Receptors can be cells, enzyme, antibody, and nucleic acid. Transducer can be optical method, electrochemical, physioelectric, and thermal, and the signal output which we can get in the smartphone or smartwatches and the PCs also. So, if we have invasive techniques already available, why we require the non-invasive method? The first important point is the available invasive methods are painful testing. Like in diabetes, also we have to prick a needle daily. So, non-invasive invasive methods can be used as a painless testing, and because of this it can provide the patient comfort, and it can tell us the early stage of the diseases, and real-time monitoring also happens. So what are the application of the non-invasive or wearable sensors? So wearable sensors can record the long-term physical activity, also biochemical parameters we can know. And as we know, wearable sensors are in the form of smartwatches, patches, or cloth-based sensors. So it can be easily carried out anywhere at any time, and point of care testing is available. So why this weight biomarkers? Because wearable sensors are more compatible with weight and which can be non-invasively detect various disease conditions. So why flexible biosensors we require? So when we want to wear the device whole day, then it should be compatible with our skin, and we should feel comfortable. That's why we require the flexible biosensors. So next is the weight biomarkers. So these are the various biomarkers which are present in this weight, which can help in the diagnosis of various disorders. So these are the disorders which we can target through this weight. Coming to the first chapter, here we have developed the miniaturized biosensor for lactate detection. As we know, lactate is the significant biomarkers in various diseases. Increase or decrease in level of lactate can cause various diseases like lactic acidosis, asthma, tissues damage, and many more. And surprisingly, the level of lactate in sweat is more than in the human serum. So this can be the better way for the non-invasive detection. So the key feature of the device is we can detect the lactate concentration non-invasively from the sweat. Here we are using the enzymatic approach, which can increase the sensitivity and specificity. The used electrochemical detection is rapid, cost effective, and we require a very small amount of samples. And the limit of detection is 1 millimolar. So this is the step-wise application flow for the functionalization of this lactate sensor. So here we can see first we take the electrode surface and then we do the layer by layer step-wise fabrication for the immobilization of lactate oxygen. First here we can see the MWCN to be used to just to increase the surface area, which will increase the sensitivity of the sensor. Then for the cross-linking of the lactate oxidase, we have used the ADC-NHS cross-linker and then immobilize the lactate oxidase. Then we have done the study. And this is the this is the electrochemical characterization which we have done just to confirm the modification is done or not. So in this graph, electrode modification graph, we can see for the Bayer and with MWC, the response was very sharp increase in the peak current. So that means it can help us in the sensitivity of the detection. Again, the enzyme immobilization data, here we can see when we use the layer by layer immobilization approach, it tells us that the lactate oxidase or any enzyme is modified over the surface or not. So here we can see with Bayer the response was less, with MWCNT again it increased, and with EDCNHS also it increased. But when we modified the lactate oxidase or immobilized the lactate oxidase over the surface, the current value was dropped down because of the lactate is the non-conductive enzyme with a non-conductive nature, and because of that it prevents the flow of ions. Moving forward, then we have done the different concentration effect. In that, from 0 to 20 millimolar concentration we have detected, and you can see in this graph with increasing concentration the peak current was increasing, and we got the linear fit with R square value of 0.98. Again, for the testing the specificity of the sensor means if I want to detect the lactate, that sensor should detect the lactate only, not particular other biomarkers like glucose, creatinine, or urea or uric acid. We have done the interference study, and here also the percent error was very low. That means our sensor is particular specific toward the lactate. Then we have done the reproducibility study just to check if we are fabricating the electrodes in the pulp, all the electrodes should give the same response. It should not vary much. So here also we can see there is a no more percent error in this graph. So it is reproducible also. Then again we have done the real sample analysis, and here also you can see the recovery percent was 99 to 100%, which means that the analyte is not decomposed or degrading in the reaction. Coming to the next, here we have developed the biosensor for the detection of glucose. As we know, glucose is the very common biomarker for diabetes mellitus disease. So the key features of this device is it is highly sensitive and specific. Also, non-invasive detection is available, and it can be flexible and variable, and the LOD is 10 micromolar. So when we talk about the glucose sensor, there are the various generation of glucose sensor. There are 4 generation of glucose sensor: generation 1, generation 2, generation 2.5, and generation 3. In the first generation, if we see this reaction, glucose converts into gluconic acid with the help of glucose oxidase enzyme. So here the oxygen is playing the important role. In the first generation, the oxygen taking place the important role in the catalyzing the reaction. And because of that, the disadvantage was we used to depend upon the oxygen concentration in our blood sample or the spit. And if it is increasing or decreasing, the percent errors was increasing. So because of that, the second generation is came into the exist. Here the oxygen is substituted with mediator redox redox solution. So the transfer of ions was happening because of this mediator redox solution. So with the generation 2, with the replacement of oxygen with mediator solution, we are able to achieve the reproducibility of the sensor. But the problem was when this solution was in the reaction, there were some free ions which were affecting the result of the sensor, and due to these free ions available in the solution, we were not uh, able to use this sensor for the implementable devices. So then again 2.5 generation came, which was similar to 2.5 second generation. Only here the mediator layer was directly attached toward the electrode surface. So there are no such free ions are available. So this generation of electrode sensor, glucose sensor, can be used for the implantable devices also. And in the third generation, the electron transfer is directly from the electrode surface. So here the electrode itself is helping in the transfer of ions. So these are the different generation of glucose sensors. So in this study, we have used the 2.5 generation. So here, for the redox mediator layer, we have used the Prussian blue, which is commonly used as a redox mediator. And after that, we have done the MWCNT, cross-linker, and glucose oxidase immobilization. So the Prussian blue was electrochemically deposited, and if we can see in this reaction that when the Prussian blue was deposited on the electrode surface, we see the significant peaks over the in the electrochemical sensing. Again, here also we have done the immobilization study just to see the layer by layer approach when we use for the immobilization, it is happening or not. And also here also we got the better result with increase in the layer because the current value was decreasing because of the immobilization of glucose oxidase on the electrode surface. Next, also we checked with EIS study. So EIS is particularly used for the study the interface property of the sensors, and also the kinetics we can know with EIS. So next, the scanned study was done. So here we can see that with increase in concentration, the current value was increasing. So with this candidate, we will get to know our reaction is diffusion controlled. Here also the concentration study which we have done from 0.01 millimolar to 2 millimolar, and here also we got the significant R square value which is 0.975. Again, then repeatability study was done just to check if we reuse the sensor, what will be the difference in the result. So here also we got the better result. There is no such kind of difference we can see with increase in the number of cycles or number of repetition of the electrodes. Next, specificity study was carried out, and here also we can see the percent error was very less. So here also we can see the sensor is particularly testing the glucose only. Then reproducibility study was carried out to check if we reproduce the electrode with same condition, is there any variation in the result? Here also we got the better result, and there is no variation. In the real sample also, we can see the percent recovery was 92 to 105%, which is quite obvious. So here also we found that there is no percent loss in the analyte concentration. Next, we have moved for the simultaneous detection of glucose and lactate. So here you can see in this graph, in this peak, the two working electrode was inserted in the same sensor with one counter and one reference electrode, and these two working electrodes was modified, one with lactate oxidase and one with glucose oxidase and glucose. And we can see the peak for glucose and lactate. We got the same peak which we got for the separate study for glucose and lactate. Same we have done the study for the glucose detection from H9C2 cardiac rat cell culture medium. So what happens when these cells cardiac rat cells grows? They consume the glucose from the media and produce the lactate. So when they are growing, when the time is increasing, that glucose concentration gets decreases and lactate concentration get increases. So that we checked here with electrochemical response. So here in the first graph, you can see with increase in time, the response was decreasing, means glucose concentration was decreasing because the cell concentration was increasing. And the reverse trend was observed in the lactate. With increase in concentration of cells, the lactate concentration was also increasing. So these results we have validated with hemocytometer. And here also we can see with increase in incubation time that the cell concentration was increasing significantly in the case of glucose and lactate. Next, here we have developed the immunosensor for the detection of CRP. As CRP is the most important biomarker for the various disease detection like cardiovascular disease, infectious disease. So here the approach used is immunosensor. So antigen antibody approach we have used, and the electrode we have used is the gold electrode on the polymer substrate. So gold, why particular gold is because they have a good binding affinity uh, for antigen and antibodies. So here also we have used the cross-linker for the immobilization of antibody. Then we have immobilized the antibody. Then the other surface was blocked with PSA for non-specific to avoid the non-specific reaction, and then with CRP, which is the antigen, and then we have done the study. So here also we have done the immobilization study with layer by layer approach, and here also if we can see with increase in the layer, the current response was decreasing. That means the this particular antigen and antibody was capturing over the electron surface. Then just to optimize the antibody concentration, we have done the multiple tests and multiple repetitive experiments. So here in this graph, you can see after the 20 microgram per ml, the response was slowed down and it was constant. So this concentration we have used for the further studies. Then also how much time we should incubate the antibody. For that also we have done the optimization study. And here also after 30 minutes, the response was decreasing. And then the concentration effect was done just to calculate from how much concentration range it can detect this particular immunosensor. In the last, this is the particular device for pathogen detection which we have developed. So as traditionally, if we go for grow into the clinic and want to know which bacterial infection is there, it takes 24 to 48 hours for the detection, and also the techniques are lab based. So here we wanted to develop a device which would be completely portable and it can detect the any bacterial infection within a 6 hours of time. So the key feature of the device is here we have used the microfluidic channel for the storage of bacterial culture, and the temperature which we have provided is with the flexible heater which act as an incubator for this bacteria culturing. So we don't require the bulky incubator, and continuous monitoring of growth can be possible with here, and the theoretical limit of detection was 0.35 CFU per ml. So this is the process flow for the fabrication of screen printed electrodes. So first we have taken some substrate, and there we have adhered the PVC sheet. Then with the use of CO2 laser, we got the mark. Then after that we applied the ink and we got the screen printed electrode. So then we have done the electrochemical detection, and in this growth study, we can see that it increased in the incubation time that current value was decreasing. Why this is happening? Because when the bacteria was growing, it was forming some layer over the electrode surface, and because of that, it was restricting the flow of ions
Sonal,
or
sorry
electrons.
to interrupt, Sonal. You have about 1.5 minutes left. You should wrap up now.
Okay.
Yeah? Yeah.
Again, the interference study was carried out with different bacteria, and here also we got this good result with E. coli. Then the concentration effect was carried out and the calibration plot was plotted, and the real sample analysis was carried out with tap water and fruit juice. So this was the device which we used, and further the antibiotic susceptibility testing was carried out with this device. And here also we can see that with increasing time, the concentration of antibiotics was increased. So future direction can be for variable biosensing applications. The development of variable system with inbuilt readout mechanism can be the better option, and same for the pathogen. Thank you. Thank you, Biosense, for the opportunity to present my work.
Thank you, Sonal. All right, we'll move it on to the mentors. Ram, Vinita, Amit.
I could go first again. Uh, yeah, I had a couple of questions. One was, I may have missed it, but was in case of your wearable sensors for glucose and um, what was the other one? Glucose and
Lactate.
lactate through sweat. Did you benchmark the measurements that your sensor is doing with, gold standard tests for the measurement of the same concentrations?
Yes, we have done the HPLC analysis, and there we have done the same analysis for the glucose and lactate. But
Okay,
I
and
didn't put the comparative result here.
I think that would be essential for you to showcase to show that your sensors are accurate. Ultimately, if the sensors are not accurate sensors, what's the point? So that was one. The other thing is, again for detection, you were using rat conditioned medium, right? Why just not?
Yes.
Why did you not just use sweat? Sweat is so easy to produce.
Yes, we have done the both with sweat also. We have done, and actually we wanted to know the concentration of lactate and glucose in the cell culture medium also. So that's why we have done both analysis. In the sweat also we have done, and in the cell culture medium also we have done.
Okay. These were the only two questions I had. So go ahead. Thank you.
Thanks,
Good luck.
Ramre. Yes,
Thank you.
Amit,
Yeah.
Vinita.
Yeah, sorry, we have been probably taking too long to ask questions. My major question is, Sonal, these biosensors are to be used with sweat. And here you are saying glucose from sweat or lactate from sweat, but so many things change the output or the composition of, I should say, composition of sweat.
Yes.
In summer, in winter, in some people drink too much of water and sweat a lot, some don't. So did you consider any of this, and what are the so-called normal levels of lactate, glucose in in sweat?
Yes.
Did they mention that, or I didn't get it?
Yes. So lactate level in the sweat is varies from 10 to 20 millimolar.
Okay.
And the glucose level is from 0.01 to 2 millimolar.
So your detection limit is falling within that.
Yes, within that. And for your second question, so we are also targeting those factors which are affecting the sweat rate and sweat composition. So our aim is to integrate with the electronic circuit, and in that circuit, so there we are setting our baseline. So in that baseline, the normal value will be there. If in any spike.
No, if I'm suffering from a cardiac problem, for example, where will you get my baseline?
It will depend upon normal range of the
whose normal range. So before I develop cardiac problem, I had say X level of lactate, hypothetically. I develop cardiac problem and then it becomes X plus Y. But we have no access to X today when I come to you or whoever for diagnosis. So what is the basis on which you are determining normal? Because that normal would vary between individuals, and your aim is to use these biosensors for the population.
Yes. So this study we have not done it, but
no, I'm asking for your thoughts. What issues will you take into account?
Actually, we are targeting the whole population. So in that, if the concentration is increasing with the person to person, so in that condition, we can just as if the normal range of lactate is 10 to 20 millimolar. So this we can take as a baseline for every individual, and with spike, with any simple or 0.1 spike in this lactate level, when it varies from person to person, that can detect in the sensor.
Okay, thank you.
Sounds good. We can move on to Amit.
Right. So this is more on the lines of what has already been kind of like as what Vinta was kind of like. I was more wondering about the detection of the bacterial or the microbial. What is the linearity? What is the linear range that it can detect, and if it would be relevant for the practical applications? Could you share some insights into that?
Yes. So for bacterial detection also, if you can see from the calibration plot is here. So it can detect from 8 to 12 CFU per ml, but we can vary the range also. So this is particular range, normal range we have taken for E. coli sensing and E. coli infection only. And the lower limit of detection is 0.35 CFU per ml. So We have done the testing in the tap water also, where
So how
the
well does
capsule.
it fit in the real world sample? I mean,
Yes,
does,
so yes, it is fitting in the real world, because when we go to the hospital and check for the bacterial infection, it takes more time, and the same concentration we can detect in our device also, and
and
the
how
time
long
is
does it
also very
take
less.
to detect it in your
Yes, it
device?
takes only 6 hours to detect the bacterial concentration.
Okay, and is that significantly different than the conventional way that people are
Yes,
regularly doing it?
conventional method takes 24 to 48 hours, and also this device is point of
That's
care
culture
device.
based,
So
right?
yes, so also the heater and everything is integrated with this device. So we don't require the incubator, bulk incubators and other conditions. So only we have to drop the samples and we get the result.
So essentially, you mean to say that you can extend or adapt your things to the point of care
Yes,
diagnostics that you can kind of like
yes.
really.
The our aim is for the antimicrobial resistance. So for that,
Right.
first we have just to develop the hypothesis, we have done the bacterial detection, and now we are moving for the antimicrobial resistance also. So we are targeting 6 pathogen, escape pathogen, for the detection of antimicrobial resistance.
Right. And if there were different bacteria in patients, would that kind of like really affect to it?
Yes. When we are saying for multiplex sensing, so there
Yes,
the
for
6
example.
pathogen will be detected.
Hmm.
So the 6 sensors are there, and particular sensor is for specific for particular bacteria. So if we are saying for 6 pathogen, one is E. coli, Streptococcus, and other Pseudomonas and all these things.
So the interference wouldn't be affecting the accuracy of your.
Yes, because that's why we have particular specificities there. The working electrode is modified with particular antibody antigen, so it will detect the particular bacteria only.
Right. And if I understand correct, there are three systems, right? There is a reservoir, there is a thermostat, and there is a temperature controller. That's all
Yes,
it kind of like
yes,
really
yes,
takes for you. And that is a compact. Do you have a
yes. We have now. We are just moving forward for compacting all these things in the single platform.
great. That might have a good application to it. Good luck to you. And thank you for presenting.
Thank you.
Thank you. That should be all.
Thank you, Sonal. Thank you, all three of you. Sonal, very nice talk. Happy we made it work. All's well that ends well. All the very
Thank you.
best. Yeah. All right. So you can stop sharing. And our next speaker is Akshata. Akshata, I see your video on, which is great. Yeah. And you
I'm
can
gonna
now
share
start.
this, guys.
Please do that. Yeah.
Thank you.
Go ahead. All the best. 20 minutes. Huh?
Thank you.
Time
Thank you.
starts when you
Yeah.
start your slide share. Great.
Okay. Good afternoon, everyone. Thank you, India Bioscience, for giving me this opportunity to present my PhD work. So, I am Akshata, a PhD scholar from JSS Medical College, Mysore. I did my PhD under the guidance of Dr. Divya Kumar at JSS Medical College. So, my PhD work was to elucidate the mechanistic role of ATF, that is apoptosis antagonizing transcription factor, in the treatment of metabolic dysfunction associated steatotic liver disease, MASLD, or previously called as NAFLD. I'd like to start with a brief introduction to MASLD and ATF. So MASLD is a highly prevalent chronic liver disease and is a leading cause for the liver transplantation. So it is typically the accumulation of fat in the liver of people who drink little or no alcohol. Typically, it's a lifestyle-driven hepatic manifestation of metabolic syndrome. It is very well established that MASLD is now linked to cardiovascular disease, chronic kidney disease, and extrahepatic cancer, making it a more complex condition. MASLD is an umbrella term that encompasses a spectrum of disease. It starts with a simple steatosis, where we see the accumulation of fat in the liver cells. When there is more than 5% of fat, we call the condition as MASL or fatty liver. The continued accumulation and other insults like ER stress and mitochondrial dysfunction would lead to inflamed liver. The condition is now called as MASH or metabolic dysfunction associated steatohepatitis. This inflammation would further scar the liver, leading to fibrosis and then hardening of the liver, cirrhosis, and finally to the end stage HCC. Sometimes MASH can skip the cirrhotic stage and directly enter the oncogenic signaling and lead to the HCC. Globally speaking, MASLD is known to affect around 30% of the world population. And coming to the India, about 38% of adults are now prevalence of MASLD in adults is about 38%, while in children is about 35% with a rising pediatric obesity. If the prevalence is so high, then what are the risk factors for that? So the main risk factors include obesity, type 2 diabetes mellitus, insulin resistance, dysbiosed gut, several genetic factors, metabolic syndrome, age, and extrahepatic complications like cardiovascular disease, chronic kidney disease, and metabolic syndrome are also interconnected with the MASLD progression. This sets the stage for me to explain the pathogenesis of the disease. Being heterogeneous, MASLD follows a multiple head hypothesis, meaning that multiple factors contribute simultaneously to the disease progression. So the intake of high fat, high sugar diet and a sedentary lifestyle where no physical exercise is seen, so our metabolism is overworked. Increased lipid accumulation and decreased lipid oxidation would lead to the accumulation of fatty acids, free fatty acids and triglycerides in the liver cells. So this leads to the steatotic form or fatter liver. So further, continued accumulation of fats would lead to the lipotoxicity, and then ER stress, metabolic stress would all lead to the inflammatory insult that the stage is called as steatohepatitis. The continued inflammatory insult would lead to the scarring of the liver, and then the disease is now called as fibrosis. At the fibrotic stage, liver can either go to the cirrhotic and HCC. So at both the stage, liver transplantation would be the only possible treatment left at the end stage of the liver. MASLD is more of a multisystemic disease where we see the interorgan crosstalk between adipose, gut, and liver axis. Briefly about ATF, the gene I worked in my PhD is apoptosis antagonizing transcription factor. So this is a transcription factor which acts as an adapter connecting different transcription factors to a common transcription machinery. Having a leucine zipper and nuclear localization signal, there is a transactivation activity of ATF. ATF has both pathological and physiological functions. Physiologically, it is implicated in transcriptional control, cell cycle checkpoint control, and ribosome biogenesis. Pathologically, ATF is implicated in triggering DNA damage response, cellular stress response, and also cancer progression by inhibiting apoptosis and also promoting cell survival and proliferation. ATF is implicated in several cancers like hepatocellular carcinoma, breast cancer, Wilms tumor, leukemia, osteosarcoma, etc. ATF and MASLD. Need for the study. Due to the increased health burden of MASLD, and since there are no globally approved drug to treat the MASLD, there is an increased demand to evaluate multiple drug targets for the disease. So, a previous study by Dr. Kumar et al. Identified ATF in the NAFLD and HCC progression. So this study showed that obesity induces a pro-inflammatory cytokine called TNF-alpha, which is responsible for the upregulation of ATF via SREBP1. The study only showed that ATF drives HCC via STAT3 and MCP1, but it did not show the role of ATF in MASH. So our overall hypothesis was to investigate the mechanistic role of ATF in the pathophysiology of MASLD. For this, we embarked on a meticulous journey by using different inhibitory approaches to understand the role of ATF in the MASH and MASH-HCC. We started with a natural compound curcumin, and then we moved on to the pharmacological inhibitor of an upstream of ATF. And finally, we went for the gene-specific liver-specific silencing of ATF to understand the role of ATF in the MASLD. With the given time, I would like to focus more on the gene silencing objective and with a brief introduction to the other two approaches. Initially, we started with the in vitro steatotic model by treating the cells with the free fatty acids and then checked for the expression of ATF. and found that ATF was upregulated in the in vitro steatotic model. Further, diet-induced animal models of MASLD was created by feeding the mice with the Western-style, high-fat, high-cholesterol diet and a sugar water. Liver tissues were collected at different stages of the disease, MASL, MASH, and HCC, and we checked for the ATF expression, which were found to be upregulated in all the stages of MASLD, MASL, MASH, and HCC. Further, we collected the leftover biopsies from the patients of MASLD and examined for the expression of ATF, and then found that ATF was overexpressed in the human MASLD. Thus, ATF is highly expressed in the in vitro, in vivo, and human MASLD. As a first approach, so we started with the natural compound curcumin. We used a preclinical model of MASH HCC for 24 weeks, which closely resembles the human MASH HCC in terms of physiology, histology, and molecular, biochemical, and transcriptomic signatures. So when we treated the curcumin at the MASH stage, we found that most of the MASH, fibrotic, and HCC features were downregulated upon curcumin treatment. Mechanistically speaking, curcumin downregulated the ATF expression, and this expression was downregulated by KLF4 and SP1 signaling pathway. Curcumin showed an epigenetic regulation of KLF4 by increasing its expression by demethylating the CpG islands. The increased KLF4 decreased the SP1, which is a direct regulator of ATF. Thus, via KLF4 SP1 pathway, the curcumin showed its ameliorating effects on MASH HCC via ATF. The second approach was to target the ATF pharmacologically. Since there were no commercially available inhibitor for the ATF, we sought to explore an upstream molecule which could be targeted for the ameliorating effect. So we knew that TNF alpha was upregulated in MASH and HCC. However, there is a TNF alpha converting enzyme which is required for cleavage of pro-TNF alpha, which is an insoluble form found on the membrane of Kupffer cells. This was released by the cleavage of TACE into soluble form, which is required for the effect of pro-inflammatory on the hepatocytes. So when we knew the relationship between the TACE and TNF alpha, we thought to target TACE with the marimestat. Marimestat is a drug that directly targets the TACE enzyme. So when we inhibited TACE via marimestat, we saw that MASH HCC was improved. And then we also saw that this inhibition was via ATF, and it also reduced the inflammation, fibrosis, and oncogenesis in the MASH HCC. Coming to the major objective, when we wanted to check the specific effect of ATF on the MASH stage using an in vivo model of MASH, we used a diet-induced MASH model for 25 weeks when we fed the mice for 12 weeks with chow diet, normal water, or Western diet sugar water. And at the 12th week, mice developed MASL, that is fatty liver. This is when we altered the liver gene expression by silencing the ATF specifically in the liver by the help of AV8, where siRNA construct that targets the ATF was incorporated into the AAV8 via the TBG vector, which is the liver-specific tyrosine binding globulin vector to target specifically liver. So this adeno-associated virus was injected into the livers via tail vein of the mice at the 12th week, and mice were monitored for the next 12 weeks. At the end of the study, we performed glucose tolerance test and insulin tolerance test, and upon sacrification, we used the serum for biochemical analysis, and then histology, molecular analysis, and transcriptomic and metabolomic analysis was carried out. When we gave the mice with the AV8 siATF, we found that the silencing was majorly in the liver, with almost 90% of the gene expression was reduced, and it was specifically in the hepatocytes compared to the non-parenchymal cells. With this preliminary pilot study, when we started the actual study, we found that hepatic silencing of the ATF reduced the body weight, liver weight, and also adipose tissue weight. When we checked for the glucose tolerance test and insulin tolerance test, we found that silencing of ATF in the liver improved the insulin sensitivity in these mice compared to the control. However, we did not see any changes in the chow diet mice with or without ATF. So the silencing of hepatic ATF ameliorates obesity and also insulin resistance. Further, we checked the serum levels of liver enzymes ALT and AST and also lipid profiles, cholesterol and triglycerides in these mice and found that ATF silencing reduced the levels of liver enzymes and lipid profile in these mice compared to the control. And more interestingly, when we looked at the liver morphology and histology, we found that upon ATF silencing, we saw a huge reduction in the lipid droplets in these mice liver tissues compared to the control, which was also supported by the oil reduced staining. And Massillary activity score is represented as a culmination of 3 different histological like steatosis, inflammation, and hepatocyte ballooning. And when we checked for these parameters, we found that ATF silencing significantly reduced these histological parameters by improving the score. So the silencing of hepatic ATF prevents liver injury, improves steatohepatitis. Further, we wanted to check the liver-specific molecular pathways that ATF regulates in the MASH. For this, we performed RNA sequencing and untargeted metabolomics in the liver tissues of all the 4 groups. And we found that the RNA sequencing analysis showed that the genes responsible for the cellular stress induction in the MASH were downregulated in the ATF silenced mice compared to the control. We also validated the key ER stress markers and found that all these markers were downregulated upon ATF silencing. Similarly, inflammatory response was downregulated and also fibrotic markers were also downregulated upon ATF silencing, thus elevating the cellular stress, inflammation, and fibrosis. What was intriguing for us was that ATF was previously known as an oncogene, which was involved morely in the cancer progression. But what was interesting for us was the oncogene that was responsible for the cancer progression showing a significant reduction in the lipid accumulation in the MASH. So this led us to explore the lipid metabolic pathways under the ATF silenced mice. And we found that the genes responsible for the lipid metabolism were significantly downregulated upon ATF silencing, and all the fatty acid synthetic genes were also downregulated upon ATF silencing. Moving further for the untargeted metabolomic, when the differentially expressed metabolites were enriched against the cake database, we found that glycerophospholipid metabolism was significantly upregulated in the ATF silenced mice compared to the control. With this, keeping only the lipid metabolites into the focus, we enriched the metabolites against this lipid subclass and superclass in the cake database and found that all the metabolites coming under the glycerophospholipid metabolism, like phosphamines, were all upregulated in the silenced mice compared to the control. Thus, the effect of silencing of hepatic ATF was through the glycerophospholipid metabolism upregulation. This was also supported by the transcriptomic analysis, where we found that the genes responsible for glycerophospholipid metabolism were upregulated in the silenced mice compared. When we enriched against the glycerophospholipid metabolism pathway, we found that all the catabolic pathways were downregulated, while the anabolic pathways were all upregulated in the glycerophospholipid metabolism. So this means that the ATF silencing favors the synthesis of glycerophospholipid metabolism and inhibits its catabolism. Further, when we did the network analysis of this glycerophospholipid metabolism, metabolites with the clinical variables like anthropometric variables, lipid profile, liver enzymes, blood glucose, insulin, we found that in the network analysis, all the metabolites belonging to the glycerophospholipid was strongly positively correlated with a strong negative correlation to all the clinical variables, indicating the significance of glycerophospholipid metabolites in the MASH progression. Further, when we subjected these differentially expressed metabolites into the small molecule pathway database, we also found that along with the glycerophospholipid metabolism, beta oxidation of the fatty acid was also upregulated in the AATF silenced mice. This was also supported by the upregulation of fatty oxidation genes that we validated on bench. Thus, the silencing of hepatic AATF promotes beta oxidation of the mice. Further, we wanted to check the interacting proteins of AATF that is involved in the lipid metabolism to exactly know how AATF is involved in the lipid metabolism in the MASH setting. When we did the interactive analysis of AATF interacting proteins with the transcriptomic signature of the AATF silenced mice, we found that ATF is involved in the positive regulation of fatty acid metabolic process via CD74 and MAPK9. For this analysis, we used two different databases, BioGrid database and Sting database, and combined the common interacting proteins that are present in both the database, and then found that it was through CD74 and MAPK9, ATF was involved in the lipid metabolic pathway. Thus, the key findings were that silencing hepatic AATF in the mice will prevent the Western diet sugar water induced steatosis, steatohepatitis, and fibrosis. It also improves the key cellular abnormalities like ER stress, inflammation in the mice. This effect was due to the increased glycerophospholipid metabolism and also promoting the beta oxidation of fatty acid in the mice. So the entire summary of my PhD research was that ATF is upregulated in the in vitro, in vivo, and human mast cell D. When we targeted the ATF with curcumin, we found that curcumin suppressed ATF expression via KLF4-SP1 pathway, and TACE inhibition by marimastat, an upstream of ATF. It prevents ATF-mediated inflammation, fibrosis, and oncogenesis in the MASH HCC. When we specifically targeted ATF in the liver, we found that the ATF silencing prevent Western diet sugar water induced steatosis, steatohepatitis, and fibrosis. And this effect was due to the increased synthesis of glycerophospholipids and also fatty acid oxidation in the mice. Thank you, and I'm open to questions.
Okay, thank you, Akshita, for the amazing talk. We can move to expert mentors for suggestions and feedback.
Hi, Akshita, very nice work, and my regards to Divya, who's a friend. Akshita, my one of my questions was regarding, if I missed it in the introduction, what is the status for the expression of AATF in human mast cell D? Has that been investigated using
Yes. I mean that was the basis for the study which when we started. So the preliminary study only identified it in NASH, but we also validated across the spectrum, and then we found that AATF was upregulated in all the stages.
Was this using Indian patient?
Yes, Indian, leftover biopsies from our collaborator at different hospitals.
I see. And what is responsible for the upregulation of ATF? I think you were suggesting, was it the NF-kappa B pathway or
TNF alpha?
I see. All right, great. I'm done. Thanks, Vinita. You can
Thank you.
Good luck.
Thank you.
Thank you, Akshita, it was a very nice talk.
Thank you.
A lot of work and presented quite well. I have sort of, possibly a tangential question. You also mentioned ATF role as sort of, contributing to cancer, various cancers, including colon cancer, this, that, and the other. I forget which were the other ones. So how do you correlate your findings with carcinogenesis, for which potentially, as you implied, AATF is responsible? So is fat accumulation in other organs causing carcinogenesis? What are your thoughts about it?
Actually, ma'am, when we started with the MASH-induced HCC, hoping that it would be due to the oncogenic nature of AATF that could be driving this oncogenesis in. But then after two objectives, when we targeted the natural compound and pharmacological inhibition, we were also seeing the decreased fat accumulation. So that's when in the last objective, we only went for the MASH. There is
I understand your data. Data are nice and clean. I'm asking you to speculate. In the sense, first question in that context is, where else is AATF expressed? All the cancers that you showed, 3, 4, 5 cancers on one of the slides, is AATF expressed in all of those tissues normally? Is it expressed more abnormally, higher levels in those cancers? And then what, based on your data on liver, is potentially a mechanism by which such cancers are also triggered? Speculation. I know it is not your data, but essentially I'm asking you to think.
Yes, if I have to think in the lipid metabolic pathway context, I would say, I can say based on what data I have, that maybe it's due to the accumulation of triglyceride and thus lipotoxicity, and so far with the inflammation it could trigger.
Colon cancer also, for example?
No, I can only talk about liver.
I'm not asking you about liver. I'm asking you about the others. If you don't know, forget it. I will just stop there. As I said, I was curious and I thought you might have read something more about this.
Mostly apoptosis antagonist is what the ATF is studied in the context of. So it inhibits apoptosis in all the other cancers.
Okay, thank you.
Thank you.
So, right, Akshita, I mean, that was a good presentation. That was a lot of like work that has been done. But as my general comment onto that, I found a lot of correlative analysis. For example, you find that there is an upregulation of AATF. Ramra has already asked, like, what I mean, you said that there is an NF kappa B that is involved in it, but I did not see. Maybe you can check what is the conclusive direct link that NF kappa B leads to the upregulation. It sounded more of like correlative, that A, you're finding NF kappa B to be altered, and B, you're finding an upregulation. But is that just a correlation, or if it really forms the mechanism of it? Would you?
No, I mean the how AATF was upregulated was already it was established. It was via TNF alpha SRABP1. It's a mechanistic study which was already done and was the basis for me to explore it in the context of MASH.
Okay, so that has already been kind of like established. But did you see it in your system as well? If this is because I didn't find that this holds true in your system as well.
Yes, sir.
Okay, so that was okay. I kind of like missed out on that. So on the same lines, when we are talking about the silencing of the AATF, so I see that it is involved in steatosis or inflammation, liver injury. I mean, you name it, fibrosis. You kind of like mentioned several of like those. But I'm still not sure what would be the mechanism or relating them together. For example, what I would imagine that AATF silencing would lead to a global metabolic profile alteration, right? You mentioned about the glycerophospholipids, so wouldn't there be other metabolomic pathways that will also be involved? What really makes you think that it is the glycerophospholipids? I understand glycerophospholipids may be involved, but is there the direct underlying mechanism that that links the AATF silencing to the phenotype that what you have kind of like described?
Yes, first of all, I think we only knocked down ATF in the liver. So to check especially why in the liver the steatosis is getting reduced. So I mean, we wanted to check the downstream of how ATF is involved in the lipid metabolism. So that's when at the last when we did this interacting analysis with the different interacting partner and the other transcriptomic signature, we did not find the direct effect on like a binding partner or something like that, because as I said, ATF majorly acts as an adapter connecting different transcription factors to the machinery. So it could be an indirect effect.
No, I was kind of like more into the silencing of it and the phenotype, which is where there is no AATF when you have knocked it down. And you see that there is a glycerophospholipids which are kind of like and associated. That's what you kind of like reached upon to. But I wasn't very it wasn't very apparent that other metabolic metabolites were also altered. Was it in a
That
global
could be
profile?
said that there could be. We only looked into the
Yes.
lipid metabolites. I mean, we
Right.
did not check the
So that
non-lipid.
was
That
kind of
that
like
could
more
be a
correlative.
significant effect.
And
Yeah,
and
that
and
could
also
be.
about the proteins, as what you kind of like. I mean, it wasn't very apparent to me that if ATF directly is interacting with these guys, or if it is an indirect interaction between the ATF and these proteins when you show those interactors or not. So,
Yeah, it's not a direct interaction.
so
So
are you
it's
clear about it? Do you know about it that it's not, because it's an interesting piece,
yes.
but that would have a very crucial information. Um, uh, Maybe a generic kind of like thing. I was also kind of like wondering these kind of like interactions of the role of the ATF in MASLT. Does it go beyond your fatty acid, fatty liver diseases, such as non-alcoholic fatty acid liver or the type 2 diabetes? These shared mechanism holds true over there as well. Is there any data, if from yours or as what Vinita has asked, some other data which kind of like suggest a similar kind of like mode of action?
Yes, the study we did was on the non-alcoholic fatty liver disease.
Okay, right.
So yeah.
Or for the obesity or type 2 or something?
Yes,
Sorry,
I think
yes.
yes.
Oh, it is applicable in there as well?
Yes.
So is that the literature that suggests it, or does that your own data would be suggesting?
No, we also checked the expressions there.
Okay, you did. So you looked into the type 2 diabetes as well?
We know not exactly the type 2 diabetes, but I can say in the context of metabolic syndrome, where we checked in the adipose tissue, in that context, not exactly the type 2 diabetes.
And if you have a final question, and if you have to advise any potential lifestyle changes or any dietary modification that may delay or prevent the onset of the disease, what would that be?
I would say, of course, prevention is always better than cure, but dietary, intervention, I don't think it's one size fits all. But then I would always recommend physical exercise to be, more on par, along with the dietary
just
restrictions.
Eat less.
Eat less. Sure, but then exercise more.
Okay. Okay. Yeah. Thank you very much. Thank you.
Thank you,
That was very
Dr.
nice.
Amit.
Thank you, Akshita, for a wonderful talk. Now we'll move to our last speaker, who's Soumya. Soumya, could you please confirm that you're ready?
Yes, ma'am.
Okay. Can you start sharing your slides?
Yeah. Is it visible? Hello?
Not yet.
Actually, it's not to me. Others can confirm.
No, I can't see it either.
No, I can't.
Now?
No.
Not yet. Not yet.
No.
We can see you somewhere.
Yeah.
Oh.
Okay, just to confirm, you are going to share button na? On Zoom?
Yeah. 1 second.
Okay.
Yeah,
now it's in sharing mode.
Yeah. No, but now we see your Zoom. Okay, but we need to see your slides. Now we see your slides. Just go to slide share mode.
Yes, ma'am.
Good. Go ahead.
Hello.
Yes, please go ahead.
Yeah.
The slides are visible.
Good evening, everyone. This is Soumya from Manipal Institute of Regenerative Medicine, Bangalore. My research work is under the supervision of Professor Sudha Warrior. And today we'll be discussing about investigation of micropeptide and understanding the Wnt signaling cascade in an AMMSC derived ALS model. So firstly, we know that ALS is a fatal neurodegenerative disease characterized by the loss of upper motor neurons and lower motor neurons. Currently, there are very few FDA-approved drugs like riluzole, edaravone, and Qalsody, which has been recently approved, like in 2023. And there are like various gene mutations observed in ALS condition, that SOD1, C9orf72, TDP43, FIRS, and OPTN, etc. So there are two types of ALS, familial ALS and sporadic ALS. Familial, which constitute up to 5 to 10%, and sporadic majorly about 90% of the cases. So the pathogenesis of ALS is known as termed as multifactorial pathogenesis because neurodegeneration in ALS is contributed by a cumulative action of multiple mechanisms. Such as glutamate excitotoxicity, which leads to the increased calcium influx into the motor neurons. This calcium accumulates in the cytoplasm and activates the calcium-dependent pathways, thereby leading to oxidative stress. In mitochondrial dysfunction, the SOD1, TDP43, and FIRS proteins tend to form intracellular aggregates, and additionally, the disease can also be progressed with the release of pro-inflammatory cytokines, reactive microglia, and astrocytes. So the researches on the neurodegenerative diseases are hampered due to the lack of in vitro models to study neurons that expresses human proteins. So to counteract this, attempts have been made to differentiate stem cells into neuronal lineage by using a cocktail of growth factors. So we know that like human tissues are only available after post-mortem. So in this case, we are limited to understand the pathogenesis of neurodegenerative diseases during their early stages. So bimesenchymal stem cells, like why not? They are most accessible source of cells. They can be isolated from various tissue sources, and they have this trilineage differentiation capacity, and also they have the innate potential for neuronal differentiation. And they're not immune reactive, and the MSCs I have been using is amniotic membrane derived mesenchymal stem cells. So we get the placental tissue, and it's considered as a medical waste. There are minimal ethical concern, and all these characteristics makes them a like ideal source of cells for disease modeling. So methodology part, we have used this panel of experiments in each step for characterization, wherein firstly the amniotic membrane MSCs were isolated from the placenta. Then a specific neuronal induction cocktail was, a motor neuron induction cocktail was added for about 48 hours. Then disease model was induced by overexpressing SOD1 G938. Then again after 24 hours, it was checked the drug treatment studies were conducted. So this was the first objective. AMMSC isolation. They were isolated by enzymatic digestion method, and these cells exhibited the characteristic plastic adherence and fibroblast-like morphology. So the gene expression analysis showed CD73 positive, CD90 positive, 105 positive, and CD34 negative. So here the trilineage differentiation capacity was also tested, where it tested positive for adipogenesis, osteogenesis, and chondrogenesis. We also did an imaging flow cytometry, wherein 96% of the cells were positive for CD105, and 98.3% cells were positive for CD73. The IF results also revealed the same, wherein it was positive for CD105 and a negative for CD45. So these results indicate efficient isolation of MSCs from amniotic membrane. So next, secondly, the motor neuron induction and characterization was done. Initially, two different protocols were used, D1 and D2. We found D2 more effective based on morphological observation as well as qPCR analysis. So after induction in a span of 48 hours, they acquired the classic neurotypic morphology. The RT-PCR results over here showed they were positive for MAP2, which is a mature neuronal marker, and the MSC marker was also tended to be reducing after differentiation compared to the control, which is undifferentiated cells. And they were also positive for OLIC2, MNX, ISL1, and CHAT. These are the typical motor neuron markers. IF results also reveal the same. And here in flow cytometry as well, we found like 91% of TUGE positive cells, 91% of synapsin positive cells, whereas the motor neuron specific 77% of CHAT positive cells. So this confirmed our motor neuron differentiation. And furthermore, the disease model was established by transiently transfecting the motor neurons with SOD1 G9-3A plasmid. The transfection was confirmed by GFP images under microscopic observation. A significant overexpression of SOD1. NER1 here is also considered as a biomarker in ALS. So both SOD1 and NER1 were found to be upregulated, whereas the OLIC2, CHAT, and EAAT2 were found to be downregulated in the disease condition compared to our motor neuron cells. So here, even in the IF studies, these three are for OLIC2. Here, the CHAT differentiation. The CHAT and OLIC2 markers were positive for the differentiation and were negative for the degeneration or disease model, whereas SOD1 and NER1 were found to be upregulated in the disease model compared to our differentiation model. The Western blot results also revealed the same, wherein OLIG-EAAT2 decrease in disease model, but SOD1 has been upregulated in the protein observation as well. So we know that like oxidative stress has also been implicated as a key causative agent for neurodegenerative diseases in ALS. Like ALS, in our study, overexpression of a mutant SOD1 has been implicated by elevated LDH, ROS, and GRIAS assay, that is the nitric oxide release assay. So these oxidative stresses were upregulated in the disease condition compared to our differentiated cells, whereas the GSH, which is the reduced glutathione, ACH, which is the acetylcholine assay, and the TMRE, that is the membrane potential, mitochondrial membrane potential, were found to be decreased in our disease model. And the staining studies, TMRE staining studies also revealed the same. Also, IF study, this was stained for SOD1. IF study, it also revealed aggregation of SOD1 here in the nucleus, indicating mislocalization, misfolded, and non-functional SOD1 as a characteristic feature of ALS. So coming to Wnt signaling, in general, Wnt signaling pathway during embryogenesis, they help in maintaining cell polarity and plays a vital role in the commitment and differentiation of neuronal precursor cells. So in CNS, they modulate axonal guidance and maintain synaptic plasticity. Example, Wnt 7A and 5A. And So briefly to explain Wnt calcium pathway, when Wnt ligand binds to the frisson receptor, the disheveled and G protein gets activated. These activated disheveled in turn activates PLC and PDE. PLC cleaves the plasma membrane component PIP2 into IP3 and DAG. IP3 in turn binds with the endoplasmic reticulum and helps in the release of calcium ions. So once there is enough calcium in the cytoplasm, CAMK2 and calcineurin gets activated. CAMK2 later activates NFAT. The activated NFAT translocates into the nucleus, and as a transcription factor, it transcripts the downstream gene, and which are majorly responsible for neuronal survival, cell migration, and adhesion. So here the SIRT1 is a calcium channel marker. So here we could clearly see that our model like consistently showed us hyperregulated Wnt calcium pathway. Given the heightened activity of Wnt calcium pathway, which plays a crucial role in disease progression of ALS and particularly influencing the calcium signaling, which is a known contributor of neurodegenerative diseases. So this prompted us to explore the potential therapeutic interventions, specifically the use of by using Wnt inhibitors as well as calcium channel blockers as a way to mitigate this dysregulation. So this brings us to our next step in our investigation, where we assess the IC50 value of various different drugs, edaravone being the positive control drug. SC401 is a micropeptide derived from SFRP4, which is a inhibitor of Wnt signal, which is a natural inhibitor of Wnt signaling. Even ICTR3 is an inhibitor of Wnt signaling pathway, amlodipine and diltiazem being the calcium channel blockers. So based on the expression patterns SC401, diltiazem and edaravone were carried forward for further experiments. The expression of Wnt Wnt calcium pathway markers were found to be regulated in the treatment condition compared to our degeneration. And a clear upregulation of MN markers like MNX, chat, and OLIC2 were found to be upregulated post-treatment, and downregulation of SOD1, NER1, and caspase 378. I have put only cas3, one graph over here. So apoptosis markers were also found to be downregulated in treated condition compared to our degeneration. Also, we did ELISA for ChRNA and chat. ChRNA is a precursor for chat gene. So ChRNA, chat, and synapsin showed upregulated, restored regulation of the neuronal markers compared to the SOD1 G9-3 condition. And we also assessed the SOD activity in the enzymatic level using a kit called by Dojindo. Here also we observed upregulated expression of SOD1 G9-3, while these drugs were able to subside the SOD1 activity. We are mainly focusing on SC401 here. And also the neurite outgrowth staining was done, this being the neurodegeneration control, the positive drug, and SC401. We can actually see the neurites growing back here. So with this, next we analyze the key indicators of cellular stress, including ROS, nitric oxide release by Gray's assay, and cytotoxicity assessment was done by LDH. These three were found to be comparatively reduced compared to our G9-3A SOD1 degeneration condition. ATP, TMRE, and ACH levels were found to be restored compared to degeneration. So the rise in the use of peptide drugs has actually reshaped the pharmaceutical industry by providing solutions to challenges which a traditional small molecule drug cannot address. So peptide drugs offer several advantages, and these include heightened target specificity, potency, and resulting in fewer side effects, and their metabolites are generally non-toxic. So in summary, this study demonstrated the neuroprotective efficiency of SC401 micropeptide in a stem cell-derived model of ALS. Our results lead to further exploration of Wnt signaling, modulators, and micropeptide-based therapeutics. So moving forward, we aim to investigate the therapeutic efficiency of SC401 in vivo and explore its potential with the existing ALS treatments. So believing that this angle of our research will bring closer to find a viable therapeutic approach and make the ALS patients' life a bit more the quality of the patients will be hopefully increased. So yeah, thank you for your attention, and I would like to express my gratitude to my supervisor, Professor Sudha Warrior, whose heart and soul of this project, the collaborators and funding bodies for their support throughout this project. And thank you, India Bioscience and PhD Cafe, for organizing this seminar series. Thank you.
Thank you, Soumya, for great talk. Now we can head to our expert mentors for your suggestions and feedback.
Yes.
Hi, Soumya, Ramrai here. Very nice talk. So, just a couple of comments. One, again, I think I had mentioned this previously as well. Try to show your data in terms of individual data points. In your case, maybe cell fields or number of cells. Rather than as bar graphs, it always helps us look at the distribution. I also am a little astounded by the lack of standard error or deviation in your results. How do you manage to get something with such little error in your experiments? It's quite amazing.
Actually,
This is in all of your bar graphs. I'm just amazed at the error bars. But anyway, be that as it may, I had a question here also. For example, how does the peptide behave vis-a-vis free radical quenchers or scavengers? The principle should be similar, is it not, against the
yes,
SOD?
sir. Yes, sir.
So have you sort of benchmarked it against, say, even natural compounds like curcumin and things like that?
No, sir. Actually, we're still working on this. So far, this was the data. So
Okay.
we are still working on the SC40, like the more on the pathway mode. Like
Okay.
how this SC401 and is there any interaction between SC401 and SOD1? Yeah.
Okay. The other question was regarding the design of the cells as they were made to differentiate towards your motor neurons.
Yes, sir.
Your experiments. I think you did show immunofluorescence data as well, but in your Western blotting, for example, you're showing that the different markers are upregulated, right? So have you done any sort of comparison, even in your immunofluorescence, have you done any comparison experiments, if it's possible, to show that the same markers are upregulated in all the cells? It's not that you have different populations which have different levels of the different markers. I'm just trying to understand the heterogeneity after having done the transfection and so on.
Sorry, sir, I didn't get you.
What I meant is, for example, if you have two neuronal markers, marker A and marker B, and you have a field of cells in which you have done the transfection with the SOD upregulation of SOD G93A, and you're trying to see whether you have converted this entire field of cells into a motor neuron population. Have you seen if the same if all the cells have both A and B, or are there populations of cells with neuronal marker A and population cells of cells with neuronal marker B?
We have not done co-staining. So it was like a single, like a stain. Co-staining we have not done. Yeah.
All right. Okay. Yeah, I'm done. Vinita, you could go ahead. Thanks.
Yeah.
Good
Thank
job,
you.
Soumya. Bye.
Thank you, sir.
Thank you, Ramray. Yeah, good presentation, Soumya.
Thank you.
I also had the same question that Ramray had, that your error bars were so tiny. Your hands must be phenomenally skilled.
It usually lies between 0.01 like that. So
yeah, but so how many cells do you count? Because all of this is in vitro, right? So how many cells do you count? Because if you count 1,000 cells, of course the bars will come down. So I want to know the minimum is how much.
So for RNA isolation, we plate in a 35 mm dish. So that's generally 1 million cells. Yeah.
No. For example, I mean, since I can't read anything that your slides show, I have no idea what markers you are showing. But earlier you had shown oxidative stress, mitochondrial deaths, or even Wnt calcium pathway. In all of these, there was an error bar. Sometimes the error bar was not even visible. So it was that close. So in such situations, what is it that you were measuring?
Actually, like we do it in triplicates and
No,
we.
no, triplicates doesn't give me an answer because you are doing everything in vitro, right? So,
Yeah.
for example, if you are measuring calcineurin, I have no idea since what is the y-axis reads like relative something something, right?
Relative gene expression, fold
Relative
change.
gene expression. But if you are looking at, say, fluorescence, are you looking at total fluorescence? And is this only the triplicate of 3 small petri dishes that you are showing us?
Yes, ma'am, the latter one.
3 petri dishes.
Yeah, yeah.
So I also am amazed because even the cell growth is not necessarily identical. So cell numbers will be different and so on and so forth. Never mind. If you have done it.
That's majorly because we overexpress SOD1. So it will. So once we overexpress since the calculation wise it remains one as the control remains one. So the overexpressed cell usually shoot up to 64 and 50 above 50 folds like that.
So your control is essentially 1 with no error. Is that what I'm? So you are talking only of fold increase.
Yes, ma'am. Yes, ma'am.
I would recommend. I don't know in your field what is done as accepted as acceptable, but this fold increase actually helps you in getting phenomenally high p-value. Okay, phenomenally high p-value because because there is no variation in your control.
Okay.
Essentially. So find other ways of representing where you actually have a difference which can be considered more physiological and more acceptable. This is a broad statement. You can think about it in with the help of your mentor. If this is acceptable in your field, go ahead with that. But somehow I don't find it really acceptable because the control should have its own variation as well. That's what I feel. The second point I wanted to ask was all of this, of course, is mesenchymal stem cell differentiation and so on and so forth. Have you tried your peptide or any of these in the, say, necropsy material or some such material from actual ALS patients at all?
Not yet, ma'am. Not yet.
Is there a plan?
Applied for the ethical concern. It has got recently approved from the hospital. So yeah.
But it is a plan.
Yeah. Yes, ma'am. Yes, ma'am.
Okay. Thank you.
Yeah. Hello, sir.
Right. Hi. So I mean, this is nice. So I would get like more molecular if you kind of like more give some insights into. You mentioned the Wnt signaling pathway.
Yes, sir.
Is there a specific component of the Wnt signaling pathway which is?
Sorry, sir, I didn't
get you. Is there a
get you.
specific component of the Wnt signaling pathway which is very significantly affected in ALS that you are aware of? You mentioned about the canonical as well as the non-canonical. So, is there a convergence in the context of ALS with regards to the disease progression? Because you just mentioned the Wnt signaling.
Yes, sir. Yes, sir.
Or for that matter, even the downstream targets, which is relevant to the
calcium signaling.
So,
yeah.
Would you elaborate a little bit more into the Wnt signaling?
Sir, actually, in Wnt signaling, once the mechanism of the drug is like when SFRP, the one I have been using is SC401. It's a micropeptide derived from SFRP whole protein. So once if SFRP4 is present, it binds to the Wnt modulator directly and it slows down it down regulates the signaling pathway. And
what I was meaning to ask is with regards to the disease progression in ALS, the ALS disease progression, Wnt signaling is involved. So, is there a specific component which is known to be significantly associated with?
Sir, actually, Wnt5A, Frizzle2, and all has been like in the transgenic mouse models. These have been observed. The upregulation of Wnt5A and Frizzle. So,
and in human ALS, they have not been found to be, or they have not been shown to be any specific correlation or any specific.
Not exactly Wnt calcium, but Wnt canonical pathway in patient samples. Wnt calcium is mainly like recognized in iPSC models and transgenic mouse models. The the literature I found to be in these.
Okay. Or if I would ask, like, so there were different mutations in SOD1 and
Yes,
other
sir.
different models that what you kind of like really had. So, I mean, is there a commonality in these different alterations that underlies the mechanism that leads to the disease progression? Because these are different genes, which are and they're distinct from each other. So
Yes, sir.
I was just trying to understand about the convergence and the actual factor which is kind of underlying.
Sir, actually, the SOD1, TDP43, and FIRS have the common symptom clinical manifestations. But C9orf7 is quite different, which is deals with the chromosomal level itself.
No, I was more with regards to, I mean, so are they all affecting the Wnt signaling in a comparable way, or there is a distinctness which is associated with individual of this, whether it is TDP4 or...
I have checked with SOD1 and TDP43, sir. Others, I'm not sure. I look into it.
So is it comparable? Is it distinct?
Yes, sir. Yes, sir.
It's kind of like a bit intriguing and don't kind of like really understand that. And anything about like the stem cell kind of like, I mean, derived from the ALS model, it can be used to further understand this.
Sir, actually, there are like little to no stem cell models as such. There are mainly transgenic mouse models and iPSC derived models mainly. So stem cell models I haven't seen, I haven't come across much literature.
Okay. And those are the models that are being used for the therapeutics and everything.
Sorry, sir?
iPSC models are the primary ones that are being used for developing the therapeutics.
Therapeutics.
Primarily.
Yes, sir. Yes, sir. And to understand the signaling pathways majorly. I mean, the disease progression.
And this is not exactly kind of like my field, so I found like certain gap in understanding it. But in overall, it was very well presented. Thank you, Soumya, for presenting your work and regards to your PI.
Thanks.
Thank you, Soumya. The great talk. So now we are towards the long-awaited disclosure of this session. So we have a giveaway winner, and our giveaway winner for today's session is Kinjal Sangharajka. I hope I'm pronouncing it right. If you are in the meeting room, please confirm with a message, Kinjal. You can email us at hello@indiabioscience to claim your giveaway. And with this, we end our today's session. And I am thankful to all the PhD speakers. And I'm also thankful to all the expert mentors for their priceless suggestions and feedback. So tomorrow we are moving towards our final day of the PhD Cafe. Tomorrow we will be having two sessions, ecology and evolutionary biology and developmental biology. So we will be waiting for your presence. Do drop by. It's our final day of PhD Cafe. Thank you so much for everyone for joining.
Thank you.
Thank you.
Thank you. Thank you.
Thank you.
you.
Pleasure,
Bye.
ma'am. Bye.
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 Bye, Amit. Bye, Vinitha.
Yeah.
Bye.
Great. I mean, I was confused with your rave. M-Ray Rao.
I know that was a
I had a hard
urgent
time. I
typo.
mean, what I know is different and what is over here is
No, I haven't spliced myself. No frame shifts, nothing. I'm the same. Take care. Have a good day.
Bye.
Bye, Vinitha. Bye, Karishma.